Hello my dear lovely students. So once again welcome back to this amazing platform of Physics Wallah, where I, Ayushi Agarwal, your botany expert, is going to teach you the second chapter of class 11th, that is biological classification. I hope you all have seen the first video of living world, and today we are going to start with the second chapter, very important again from the point of view of NEET. So let's not waste time and directly jump into the topic. Now, this chapter is basically, bachcho, a kind of continuation of the first chapter itself, living world. How? In living world, you were studying about taxonomy. You were studying about what is classification, what is identification, etc. In this chapter, you are going to study that what is there in classification, what scientists did actually after classification, in what groups are those organisms divided. So in this chapter, we are going to study the different classification groups of organisms. First thing. Second question comes in the mind, that why the hell are we studying so much? What is the need of biological classification? Why are we made to study? So beta, this is very important chapter. Why? Because it is very important to know that what is the biodiversity around you. There are 17 to 18 lakh species which have been discovered so far. And is it not our responsibility to know that what organisms are present around us? Therefore, biological classification becomes very important in order to understand the vast biodiversity on Earth. Ma'am, why? Again question comes, ma'am, why? Ma'am, you are saying that it is important to know the organisms present around us, but we don't find it important. Then why are we made to study this biological classification? What is the point of classifying organisms? Bachcho, it is equivalently important for you as well. Because once you know that what are the organisms present around you, once you know the importance of those organisms, then only you can exploit, you can extract benefits out of them, either in the form of food, shelter, or clothing. Yes or no? Like for example, there is a tree. Now you know that the tree yields pulp which you can use for making rubber. How you know that tree is going to give you a pulp which you can use for making rubber? You only came to know when some scientist studied on that organism, classified that organism, identified that organism. Then only you came to know the benefit of that tree. And then only you can prepare the rubber out of that plant. So that is why once you study the organisms present around you, then only you can extract benefit out of them, either in the form of food or in the form of shelter or in the form of clothing. Hence, now I hope you must have realized the importance of classification, that to extract benefit from the organisms, either in the form of food, shelter, or clothing. Clear to everyone? So these are the two needs of biological classification. Now, owing to the needs of biological classification, many scientists tried to group organisms. The first scientific approach was done by scientist Aristotle. Aristotle, he proposed the first scientific approach of classification. Aristotle, my dear students, divided organisms into two categories. The simple two categories as plants and animals. Right? Further, on the basis of size, he classified plants also into 3 subcategories: herbs, shrubs, and trees. So he further classified plants into 3 categories on the basis of size: smallest ones as herbs, the medium ones as shrubs, and the tall ones as trees. Whereas Aristotle divided animals also into 2 broad groups on the basis of the presence or absence of RBCs. RBCs are the red blood cells. The 2 broad groups are Enyma and Onyma. Enyma means beta those organisms whose color of the blood is red due to the presence of RBCs. So RBCs, if is found in the blood cells, then those organisms, according to Aristotle, belong to Enyma, whereas means without. So those organisms in whose blood RBCs are missing, they were grouped under Onyma. So Onyma means without RBCs. This was the simple classification proposed by Aristotle. He just gave a layman kind of classification. He just gave the first scientific approach of classification. However, the first formal classification system, first proper classification system was given by scientist Linnaeus. So Linnaeus gave the first formal classification system. He actually used the term kingdom for the first time, and he proposed 2 kingdom classification. The 2 kingdoms proposed by Linnaeus, my dear students, are kingdom Plantae and kingdom Animalia. So, who gave the first formal classification system? The first formal classification system, your answer should be Linnaeus. Which is that classification? 2, 3, 4, 5? Your answer will be 2. What are those kingdoms? Animalia and Plantae. Now, my dear students, he actually did this by studying certain features. Let's see what are those features. So, features which were used by Linnaeus, the most important feature on the basis of which his whole classification system is based, is cell wall. If cell wall is found in the cells of the organism, then they are kept in the kingdom Plantae, and if cells are not found, then they are kept in the kingdom Animalia. First and foremost feature. Apart from this, he also considered many more features, like for example, ability to eat. If one is able to eat, then that organism is kept in the kingdom Animalia. If not able to eat, like trees, then it was kept in the kingdom Plantae. Another feature, ability to move. If one is able to move, then that organism belonged to kingdom Animalia. And if one is unable to move, then that organism was kept in the kingdom Plantae, sedentary, non-motile. Clear? Apart from this, he also considered one feature called response to stimuli. Response to stimuli. Let's write first. Now you will think, ma'am, in the last chapter itself, you told that consciousness is the behavior, it is the defining feature of all living organisms, and plants and animals both are living. So they both will show response to stimuli, they both are conscious type of organisms. Then how is it creating a difference? Why Linnaeus chose response to stimuli as one of the feature of classification? Actually, Linnaeus said response is given by both plants as well as animals. But if the response is slow, then slow response givers will be kept in the kingdom Plantae. And if the response is quick, then those organisms will be kept in the kingdom Animalia. Right? Yes or no? Now I hope this feature also makes sense. Clear. One more feature which he considered was the presence or absence of contractile vacuole. Beta, contractile vacuoles are found in the cells which are responsible for maintaining the water balance, that is osmoregulation. So contractile vacuoles are responsible for maintaining osmoregulation. According to Linnaeus, if contractile vacuole are present in the cells of an organism, then that organism belongs to kingdom plant Animalia. And if contractile vacuoles are absent, then that organism belongs to kingdom Plantae. So, my dear students, this was the simple classification system of Linnaeus. And according to his classification system, he kept organisms like protozoans, chordates, and non-chordates under kingdom Animalia, and organisms like bacteria, fungi, algae, bryophytes, pteridophytes, gymnosperms and angiosperms in the kingdom Plantae. Do you find this classification system good? Do you find it perfect? Then your answer will be no. Why? I hope you all can see the demerits of this classification system by your own self. How? Let me point out one by one. Number 1, bacteria. Beta, bacteria are made up of prokaryotic cells and remaining are eukaryotic. So how can eukaryotic and prokaryotic be kept in the same kingdom? This was his first flaw. This was his first demerit. Second, bacteria and many more organisms, they are single-celled. They are unicellular, and remaining majority of them are multicellular. So again, unicelled, multicelled kept together does not make a good sense. So that becomes a second reason for discarding Linnaeus classification system. Third, can you see fungi and many of the bacteria as well? Their mode of nutrition is heterotrophic. They cannot prepare their own food. But if you see algae, bryo, pterido, gymno, angio, they all are autotrophic. They all can prepare their own food. So how autotroph and heterotroph can be kept in the same group? This also does not make a better sense. So these were certain demerits of this two kingdom classification. Thus now this classification system was replaced by Ernst Haeckel's three kingdom classification. Ernst Haeckel was the scientist who proposed the third kingdom as Protista. So now the three kingdoms are kingdom Animalia, kingdom Plantae, and kingdom Protista. In kingdom Protista, my dear students, he kept all the unicellular eukaryotic organisms. Right? He removed all the unicellular eukaryotic organisms kept in either plant or animals and kept them together in the kingdom Protista. So similarly, all the protozoans which were initially kept in the kingdom Animalia, they were removed from there and they were moved to Protista. Clear? So what are the three kingdoms now? Animalia, Plantae, Protista. Protista is that group which comprises of all unicellular eukaryotic organism, and this kingdom was proposed by scientist Haeckel. Clear? Do you find this also sufficient? No, it is still insufficient because yet bacteria's prokaryotic organisms are kept in the kingdom Plantae. Therefore, three kingdom classification also got discarded and was replaced by four kingdom classification proposed by scientist Copeland. Copeland ने बच्चों four kingdom classification propose किया and the four kingdoms are Animalia, already you know, Plantae, already you know, Protista, already you know, and the fourth kingdom established by Copeland was kingdom Monera, in which he kept all unicellular prokaryotic organisms, in short bacteria. So Monera comprises of prokaryotic organisms. Again, one trouble is solved. Bacteria are now removed from kingdom Plantae and kept in kingdom Monera. Good, sufficient enough. Are you happy with four kingdom classification? Shall we stop? No, ma'am, no. Four kingdom is still not sufficient. Why? Because fungi, the heterotrophic organisms, are still belonging to kingdom Plantae, which is wrong. Hence, this four kingdom classification was replaced by R.H. Whittaker's five kingdom classification. So R.H. Whittaker is a scientist who proposed five kingdom classification. He removed fungi from the kingdom Plantae and established itself as an independent kingdom. So now the five kingdoms are, what are the five kingdoms? Can you name them, my dear students? Will you all try? Very easy. Let's write it. Animalia, Plantae, Fungi, Protista, and Monera. ठीक है? So these are now the five standard kingdoms after R.H. Whittaker classification. R.H. Whittaker बच्चों gave five kingdom classification after studying certain features, out of which the most important is mode of nutrition. He studied many features for classification, but the most important being mode of nutrition. This mode of nutrition is actually the important basis on the basis of which kingdom fungi was established. Clear? Apart from mode of nutrition, he also considered features like body organization, that is thallus organization, whether organization is organ type, organ system, tissue grade, etc. Mode of nutrition, I told you it is most important. Cell type, that means whether body is prokaryotic, eukaryotic, single celled or multi-celled. Reproduction, methods of reproduction, what is organism preferring, sexual, asexual. Then presence or absence of nuclear membrane, that whether the genetic material is true or it is false. False means nucleoid type. Then phylogeny. R.H. Whitaker, my dear students, also considered evolutionary relationship as one of the important feature of classification. R.H. Whitaker also considered phylogeny as one of his feature for classification. Right? So these are the list of the features which was considered by R.H. Whitaker during his classification. Now we are going to see his classification system in a chart manner. Right? So this chart, we are going to typically do it from NCERT, and it is very, very important. Direct questions are asked from this chapter. If you go and check the questions asked in previous year NEET examination, there's so many questions asked directly or indirectly from this chart. So please guys, all my dear students, pay attention and be sincere. So this is an empty chart which now we are going to fill. All the outlines have been done. These are the 5 kingdoms and the list of the features which was considered by R.H. Whitaker. Sabse pehle, the first one is cell type. So except kingdom Monera, all other organisms, all other members of different kingdom are made up of eukaryotic cell, and members of kingdom Monera are made up of prokaryotic cell. And if we talk about the remaining kingdom members, then they are made up of eukaryotic cells. Clear? Moving ahead. Second, nuclear membrane. Now my dear students, you can see that only kingdom Monera is having prokaryotic type of cell organization or cell type. Therefore, prokaryotic cells beta, they do not have a well-defined genetic material. Their genetic material is diffused in the cytoplasm and not surrounded by any nuclear membrane. Such type of genetic material is known as nucleoid or genophore. That means genetic material is diffused in the cytoplasm, right? But if we talk about the remaining kingdom members, so my dear students, all are made up of eukaryotic cells, and the characteristic feature of eukaryotic cell is presence of well defined genetic material. The genetic material where nuclear membrane is present, and their genetic material is known as nucleus. Agreed? Yes or no? Yes. Very good. Moving ahead. Third important feature of classification is cell wall. Cell wall beta, presence of cell wall is a feature which is found in different organisms belonging to different kingdom. If I talk about animal cells, straight away you can write that in animal cells nuclear membrane is absent. Sorry, cell wall is absent purely. But in remaining, if I talk about kingdom Monera, then in Monerans the cells are having cell wall. However, their cell wall is made up of proteins and sugars. That is amino acids and sugar. In short, the cell wall of Monera is made up of murin, peptidoglycan. Right? Yes or no? Will you remember this term? The other term for peptidoglycan is murin. So remember this thing, that the cell wall of kingdom Monera is made up of peptidoglycan. In Protista, cell wall may be present, may not be present. If present, then usually it is cellulosic. In case of fungi, cell wall is present and it is chitinous in nature. So, the cell wall is present in fungi, but it is made up of chitin. Chitin is also a polysaccharide. It is actually a polymer of NAG, N-acetylglucosamine. Right? Coming to plantae. Plantae to hai hi. So, its cell wall is present and which is cellulosic. Animals may no cell wall. I hope this feature is also clear to all my students. Moving ahead, the next feature which he considered and the most important feature of R.H. Whitaker is mode of nutrition. So mode of nutrition is very important feature and the most important feature of classification. Nutrition mein beta, according to R.H. Whitaker, Monera shows maximum diversity in term of nutrition. In case of Monera, majority of the members can be either autotrophic or heterotrophic. In autotrophic also they show different categories. In heterotrophs also they show different categories. That I will teach you when I'll teach you kingdom Monera separately. Here you can only write that mode of nutrition can be both autotrophic as well as heterotrophic. Right? And they show maximum diversity. Coming to Protista, Protista also shows diversity in terms of nutrition but less than Monera. The members of Protista can also show both autotrophic and heterotrophic mode of nutrition. Details we will do once I will teach you the kingdoms independently. Here we are just fulfilling the chart, so no further details. Further details will be done when I will teach you all the kingdoms independently. Correct? If I talk about fungi because they are heterotrophic, because the members of kingdom fungi cannot prepare their own food, that is why they were removed from plantae and established as an independent kingdom. So that is why mode of nutrition becomes a very important feature. Clear? So mode of nutrition is heterotrophic, plantae, clear, autotrophs, animalia, clear, heterotrophs. Clear? Animals cannot prepare their own food. They depend upon another sources. Therefore, they are consumers. They are heterotrophs. They are not producers. Coming to the last feature of R.H. Whitaker in this chart, that is body organization. Body organization, if I talk about kingdom Monera and if I talk about kingdom Protista, then better they are unicellular organisms. That is why we say that their body organization is cellular body organization. Clear? Single cell body organization. Moving ahead to fungi, fungi shows loose body tissue type of body organization, loose tissue body organization. So fungi shows loose tissue body organization. Plantae, proper tissue body organization, and they can also form organ like roots, stems, leaves. These all are the organs of plants. So body organization exhibited by plant ranges from tissue to organ level. So maybe tissue, or maybe organ type of body organization. Animals में तो आप they show organ and organ system type of body organization. The highest range of body organization is exhibited by animals. So you can simply write tissue, organ या organ system. Organ or organ system type of body organization. Hope this chart is clear to all and every student. Please learn this chart and questions will be on your tips. Apart from these features, one more important feature which Whittaker considered was phylogeny. He also traced evolutionary history in order to group organism. Remember that. Clear? Moving ahead, bachcho. Hope the 5 kingdom classification is clear. Now, when we come in modern era, in today's world, in today's era, after Whittaker came one more scientist by the name Carl Woese. Carl Woese, beta, he was studying, he's a microbiologist. He was studying on the members of kingdom Monera. He was studying on bacteria. He found 2 broad deviations in kingdom Monera. He found 2 broad groups in kingdom Monera showing great differences. One group of bacteria was exhibiting one type of property and another group of bacteria in the same kingdom was showing some different property. Therefore, Carl Woese thought that when in the same kingdom Monera, 2 such groups are developing, then why not to separate these 2 groups and establish them as independent kingdoms? Hence, Carl Woese divided kingdom Monera into 2 new kingdoms as kingdom Archaebacteria and kingdom Eubacteria. Hence, from 5, now we became 6 kingdoms. The 6 kingdoms are kingdom Archaebacteria, kingdom Eubacteria, Protista, Plantae, Fungi and Animalia. Clear? So what Carl Woese did? Carl Woese divided kingdom Monera on the basis of certain features which I will tell you. Divided kingdom Monera into 2 new sub-kingdoms as kingdom Archaebacteria and kingdom Eubacteria. Thus now how many kingdoms are there? 6 kingdoms are there. Those 6 kingdoms are Archaebacteria, Eubacteria, Protista, then Fungi, Plantae and Animalia. Yes or no? Apart from creating 6 kingdoms, Carl Woese bachcho, he also developed one more category of classification above kingdom. He created one more category of classification by the name domain. Till now we know that kingdom is the topmost category of classification. Yes, but Carl Woese created one more category above kingdom called domain, and he placed these 6 kingdoms into 3 domain. Thus, his classification system is known as 3 domain, 6 kingdom. 6 kingdoms you can see. Now let's see what are those 3 domains. The 3 domains are domain Archaea, in which he kept kingdom Archaebacteria, domain Prokarya, which includes kingdom Eubacteria, and domain Eukarya, in which he included the remaining kingdoms consisting of eukaryotic cells, that is Protista, Fungi, Plantae and Animalia. So therefore his kingdom classification is known as 3 domain, 6 kingdom. 6 kingdom you understood, 3 domains are over here. Domain Archaea, domain Prokarya and domain Eukarya. Correct? Will you remember? Out of 3 domains, in 2 domains, there are organisms with prokaryotic organization, and in 1 domain, there is organisms with eukaryotic organization. Will you remember this? Let me write it for you. So, out of 3 domains, you know the name. 2 domain members are prokaryotic. You know the 2 domains, number 1 and number 2. And 1 domain, that is domain Eukarya. Here the members are eukaryotic in nature. Clear to everybody? Right. Now the question comes on what grounds he did, what actually he observed in kingdom Monera that he thought to divide kingdom Monera into 2 new kingdoms. So the first and foremost thing which he observed was the gene sequencing of 16S rRNA. He was actually studying the ribosomes of bacteria. The ribosomes of bacteria are 70S type, right? 70S ribosomes, they are made up of 2 subunits, the larger 50 and smaller 30. The smaller subunit, that is 30S beta, this is made up of one of the type of rRNA called 16S rRNA. This fellow Carl Woese was trying to understand the gene sequencing, the arrangement of nucleotides in 16S rRNA. And he was surprised to find that in the same kingdom, there were 2 different results of 16S rRNA gene sequencing. One group of organisms in kingdom Monera was showing one result of gene sequencing, and there was another group of organisms in the kingdom Monera which was showing some different result. Carl Woese said that if such basic thing cannot be similar in the same kingdom, then these members are not fit to be kept in the same kingdom. And hence, on the basis of 16S rRNA gene sequencing, Carl Woese separated kingdom Monera into 2 new kingdoms. Why? Because members of kingdom Archaeobacteria were showing some different result, and members of Eubacteria were showing some different result of 16S rRNA gene sequencing. Clear? Yes or no? Yes? Okay. Very good. Very good. Now let's proceed further. Here again one more chart is there. Charts always makes the study more interesting and easy, right? So now here I am going to show that what are the features which Carl Woese considered apart from 16S rRNA gene sequencing in order to establish 3 domain 6 kingdom classification. So here I am showing the 3 domains as domain Archaea, domain Prokarya and domain Eukarya, in which the 6 kingdoms are divided. And these are the list of features which Carl Woese considered in order to propose his classification. First and the very important feature, 16S rRNA gene sequencing. So when he did 16S rRNA gene sequencing, when he tried to understand the nucleotide arrangement in this 16S rRNA, he found different results. What are those different results? We are not concerned. You should simply know that the result of 16S rRNA gene sequencing was different in the kingdom Monera. Hence, he divided kingdom Monera into two new kingdoms. Right? Clear? Moving ahead. Nature of nucleus. Apart from 16S rRNA, my dear students, Carl Woese was also studying on the structure of nucleus. He found that in members of archaebacteria, histone protein is present in their DNA, which usually is not present in prokaryotic cells. So this is an exclusive feature of archaebacteria, that histone proteins. DNA is nucleoid type. No nuclear membrane is present around archaebacteria, but histone proteins were found. However, the nature of the genetic material is nucleoid. In prokaryotes, genetic material is nucleoid, but no histones. Here, histones are found. So, can you see this similarity of archaebacteria with eukarya domain? Another feature which he considered was the structure of plasma membrane. My dear students, whether it is a prokaryotic cell or a eukaryotic cell, the only structure similarity is the plasma membrane. Plasma membrane is uniformly common in all the type of cell, and it is represented by lipid bilayer with unbranched side chain. So whether it is prokaryotes or eukaryotes, the structure of plasma membrane is same, which is lipid bilayer. You know. If you don't know, then this also we will cover in our chapter cell. So till then, just stay tuned. So this is how the lipid bilayer is. And this type of plasma membrane is found in prokaryotes as well as eukaryotes with unbranched side chain. But if I talk about archaebacteria, if I talk about the domain archaea and kingdom archaebacteria, then Carl Woese found that their plasma membrane is represented by lipid monolayer plus branched side chain. Isn't it a different factor? Isn't it quite different? And that is why this fellow, Carl Woese, decided to separate members of archaebacteria from the members of eubacteria. Clear? Coming to the next feature, cell wall composition. Normally, you have seen the 5 kingdom chart. We have written that in kingdom Monera, the cell wall is made up of murin. It is made up of peptidoglycan. Yes or no? So here, cell wall is peptidoglycan, commonly called as murin. But in case of archaebacteria, if they were true bacteria, if they belong to kingdom Monera, then their cell wall should also be made up of murin. But no, the composition of cell wall in archaebacteria is not murin. Rather, it is called as pseudomurine. I will tell you what is the difference between murine and pseudomurine. later. But here you should know that the cell wall of archaebacteria is made up of pseudomurine, whereas the cell wall of prokarya is made up of murine. Eukaryotes, depending upon the kingdom, cell wall may be chitinous, cell wall may be cellulosic, hemicellulosic, etc. So cell wall here you can write may be chitinous, may be cellulosic, hemicellulosic, etc. Clear? So there's so many differences that Carl Woese decided to divide kingdom Monera into two sub-kingdoms as kingdom Monera, as kingdom Eubacteria and kingdom Archaebacteria. So this is the latest classification system of Carl Woese, 6 kingdom and 3 domain. I hope all the classification systems are clear to each and every one of you. Yes or no? If yes, then just give a thumbs up in your comment section. Right? Okay. So, this is all about the classification system. Now, we are actually entering into the main part of the chapter where I am going to teach you each kingdom independently. First, starting with kingdom Monera. Kingdom Monera means first we are going to talk about the general features of both the subkingdoms Archaebacteria and Eubacteria, and then I will tell you their unique properties. Clear? So first we are going to start with this kingdom, and first we are going to start with their general features. Am I clear or not? So we are going to start now with the first kingdom, that is kingdom Monera. First we are going to discuss about their general features and then we are going to talk further. First of all, beta, the first and the foremost important line regarding this kingdom, you should know that bacteria are the sole members of this kingdom. This kingdom was proposed by Monera, this kingdom was proposed by Copeland, who kept all unicellular prokaryotic organisms into this. Later on it was separated by Carl Woese, but initially all the unicellular prokaryotic organisms were kept in this kingdom and bacteria are the sole members. I don't remember the year in which NEET has asked question on this line. The dash is the sole member of kingdom Monera. And so options were given and the correct answer was bacteria. So bacteria are the sole members of kingdom Monera. First and foremost important line. Bachcho, bacteria, they are cosmopolitan. Cosmopolitan means the ones which are found everywhere. On this board, on the fan, lights, everywhere, soil, everywhere you will find number of bacteria. So bacteria are cosmopolitan. That means they are found everywhere. Now, if I talk about the cellular structure of the bacteria, if I talk about the cellular structure of the members of kingdom Monera, then bachcho, their genetic material is naked type. Their genetic material is not well defined. Not well defined means that genetic material is represented. For example, this I am drawing a small bacteria. Right? These are the outer membranes of the bacteria. Right? For example, I call it as plasma membrane. Now, the plasma membrane inside encloses the fluid which is called as cytoplasm. Within the cytoplasm, the genetic material is present, directly diffused. It is not separated by any nuclear membrane. No histone proteins are found in their genetic material. Plus, the genetic material is represented by double-stranded, circular, single chromosome. And such genetic material is known as nucleoid or genophore. Right? So what is genetic material? Genetic material in Monera is represented by double-stranded, circular, single, naked. Naked means no histones plus no nuclear membrane. Right? Along with this feature, bacterial cytoplasm also do not have any compartmentalization. Compartmentalization means that there are no membrane-bound cell organelles like ER, Golgi, mitochondria, etc. They only have non-membrane bound organelles like ribosomes. So next important feature which you should know is that they lack membrane bound organelles. However, non-membrane bound organelles like ribosomes are present in the cytoplasm, and the nature of ribosomes is 70S. Clear? Yes or no? Very good. Moving ahead. Now bachcho, these bacteria, these whole soul members of kingdom Monera, their body is just represented by one cell, and that cell is also prokaryotic in nature. Correct? So their body organization is very simple. However, if we talk in terms of their functional aspects, if we talk that how do they live their life, then their living pattern is not that simple. So we can say that the simple bacteria are simple in organization but exhibit complex behavior, like their mode of nutrition, respiration, reproduction is not as simple as their structure. This line I have taken directly from NCERT, very important line. It can come either in the form of correct, incorrect, true, false, match, following, etc. So please mark this line as important. That bacteria, they possess simple organization, body organization, but exhibit complex behavior. Clear? Very good. Now, due to the absence of certain proteins like tubulin protein, the cytoplasm of bacteria do not show any movement. There are no cytoplasmic streaming taking place in the bacterial cytoplasm. Bacterial cytoplasm is still. So we can say that bacteria, the members of Monera, do not exhibit cyclosis. Cyclosis means cytoplasmic movement. There is no cytoplasmic movement taking place in the cytoplasm of bacteria. Right? They lack proteins like tubulin, so no microtubules, no cytoskeleton structure also found in the members of kingdom Monera. Moving ahead. Now, on the basis of shapes, if you study the structures and the shapes of bacteria, then bacteria can be of 4 distinct shapes. They can be either comma shaped, called as Vibrio, like Vibrio cholerae, which causes cholera. Second, they may be rod shaped, called Bacillus, like Bacillus anthracis. So Vibrio, Bacillus spiral shaped, like spirochetes, called as spirochetes. And some may be spherical in shape, called cocci. Oh, just a second. Spirochetes. And some may be spherical in shape, called cocci, like Streptococcus bacteria. Right? So on the basis of shape, bacteria can be of how many types, students? 4 types. So we can classify bacteria on the basis of their morphological appearance. Thus, bacteria are also known as morphological species, morphospecies, because we are classifying them on the basis of shape. They can be of 4 types: Bacillus, Vibrio, then your spirochetes and cocci. Right? Yes or no? Very good. Moving ahead. Some bacteria may be motile and some bacteria may not be motile depending upon the presence or absence of flagella. Remember, if flagella is found, then bacteria are motile. If flagella is not found, then they are called as non-motile. These flagella are the extensions of the plasma membrane and they are made up of flagellin protein. Apart from flagella, bacteria may also possess some more extensions either in the form of pili or in the form of fimbriae. Pili are beta made up of pillin proteins. They are helping in allying the exchange of genetic material between the two bacteria through conjugation. I will tell you what is conjugation, but till now understand that bacteria may give rise to extensions like flagella, that is responsible for motility, made up of flagellin protein. Pili, made up of pillin protein, helps in exchange of genetic material, and fimbriae, made up of fibrin protein and responsible for the attachment of the bacteria to the substratum. So this fimbriae is responsible for the attachment of bacteria to the substratum. Clear? So this is also one of the important general feature which you should know. So now you know what are pili, you know what are fimbriae. These are just the extensions of which are found in the prokaryotic cell and exhibiting some unique features. Now bachcho, in bacteria, apart from their main genetic material, the main genetic material which is known as nucleoid, genophore, I told you. Apart from that, apart from main genetic material, bacterias, the members of Monera, also possess some extra chromosomal genetic material. Some extra chromosomal self-replicating circular DNA called plasmid. Self-replicating circular DNA called plasmids. This is a unique feature of prokaryotic cells only. Plasmids are absent in eukaryotic cells. So what are plasmids? They are extra chromosomal self-replicating genetic material found in the cytoplasm of bacteria. Clear? This is also an important general feature which you should know. Now moving ahead. So these all are the general features which you should know regarding the members of kingdom Monera. Now we are moving ahead towards their diversity, towards their functions, towards their activities, what they perform. And the first one which I am going to discuss is their mode of respiration, that how bacteria respire. Beta, bacteria shows diversity in terms of mode of respiration. There can be 4 broad categories of bacteria on the basis of their mode of respiration, which I have divided as obligate aerobes, obligate anaerobes, facultative aerobes and facultative anaerobes. Right? So all the 4 types of respiration now I am going to tell you. Starting with obligate aerobes. Bachcho, obligate means strict. compulsory. So obligate means strict. Aerobe means oxygen. So those bacteria who compulsorily require oxygen for respiration are called as obligate aerobic bacteria. On the other hand, obligate anaerobe, name itself suggests obligate means strict, anaerobes means without oxygen. So those organisms who do not require oxygen, who respire in complete absence of oxygen, are called as obligate anaerobic bacteria. Shall we write it? Yes, come on. Obligate aerobes, bacteria who require compulsorily require oxygen for respiration. Anaerobes, bacteria who do not require oxygen for respiration are called as obligate anaerobes. Clear? Next set, facultative aerobes, facultative anaerobes. Facultative means not strict, alternative. So facultative aerobes means those bacteria who normally do not require oxygen, who normally are anaerobic, but if provided with oxygen, then they will also start respiring in aerobic conditions. So, what are facultative aerobes? Write it. See, bacteria who normally do not require oxygen. They are anaerobic. However, can also respire aerobically. Right? On the other hand, contradictory to this is facultative anaerobes. Facultative anaerobes are those bacteria who normally require oxygen but can also respire in the absence of it. Correct? Shall we write it? Chalo, let's do it. So bacteria who normally require oxygen, however, can also respire anaerobically. They are called as facultative anaerobic organisms. Clear to everyone? Are all the four types of mode of respiration clear? Once again, obligate aerobe, strict aerobic, obligate anaerobe, no oxygen, strict no oxygen, facultative aerobe, which require no oxygen usually but can also respire aerobically, facultative anaerobe, who are naturally aerobic but can also respire anaerobic if conditions are provided. Clear? So this is one of the mode of diversity in bacteria. Apart from respiration, next important function exhibited by bacteria is nutrition. So moving ahead to nutrition part. Bacteria, as I told you, beta, they exhibit maximum diversity in terms of mode of nutrition. This question was asked in 2011. You can go and check the paper in 2011. This question was asked that which kingdom exhibits maximum diversity in terms of nutrition? So your answer will be Monera, bacteria. Shall I write this point? Very important, highlighted. Maximum diversity in terms of nutrition. Chart yaad aa raha hai? Can you remember the chart? Can you recall that? There you wrote that bacteria can be either autotrophic or heterotrophic. They can prepare their own food, and some bacteria cannot prepare their own food. Now, autotrophic can be of two types: photolithoautotroph or chemolithoautotrophs. Photolithoautotrophs, my dear students, are those group of bacteria who utilizes sun energy to prepare their food. Photo means sunlight. Autotroph means can prepare their own food. So those group of bacteria who prepares food in the presence of light, right, and uses electron donor as any inorganic compound like water or hydrogen sulfide, and uses inorganic carbon source as well as electron donor, right? For example, purple and green sulfur bacteria. Very important, you should remember this example. Purple sulfur bacteria and green sulfur bacteria. These bacteria, they can prepare their own food by utilizing light as a source of energy, and carbon source, electron source are some inorganic compound like CO2 and water or hydrogen sulfide. Clear? Moving ahead. Chemolithoautotrophs. These are also those group of bacteria which can prepare their own food but utilizes chemical as a source of energy. Chemo means chemical. So those group of bacteria prepare their own food by utilizing chemical energy. Like for example, you see in case of nitrifying bacteria like Nitrosomonas, Nitrosococcus, they use the energy developed by the oxidation of ammonium and nitrites that you study in nitrogen metabolism. So nitrifying bacteria. Nitrifying bacteria like Nitrosomonas, Nitrosococcus, Nitrocystis, Nitrobacter, beta, they generate energy by oxidizing ammonia to nitrite and nitrite further to nitrate, and while doing this, whatever energy is produced, such bacteria utilize it for preparing their own food. And therefore, these kind of bacteria who can prepare their own food by utilizing chemical as a source of energy are known as chemolithoautotrophs. Clear? Yes or no? So, on the basis of nutrition, there are two types of bacteria. Photolithoautotroph, on the basis of autotrophic mode of nutrition, there are two types of bacteria: photolithoautotroph, chemolitho. Photolitho means bacteria who can prepare their own food by utilizing light as a source of energy. Chemolitho, bacteria who can prepare their own food by utilizing chemical as a source of energy. Right? Now, next category is of heterotrophic bacteria who do not prepare their own food. Majority of bacteria are heterotroph, and in heterotroph also there is one category of decomposers. So majority of the bacteria are decomposer type of heterotroph. Right? Let's write it. Heterotroph, bacteria who do not prepare their own food. They are basically consumers. Now heterotroph में कई सारी categories होती हैं. Some bacteria may be parasitic. Some bacteria may be symbiotic. Some bacteria may be saprophytic. But majority of them are decomposers which feed on dead and decaying organic waste. So, okay, yeah. So if I talk that which is the maximum type of nutrition exhibited by bacteria, heterotroph. And in heterotroph, if I ask you which is the maximum type of nutrition exhibited by bacteria, then your answer will be decomposer. Please note it down. Mark it as important. Question will come from this section as well. Right? So this is all about nutrition which you have to do. Now you have studied two aspects of bacteria. One is their respiration, another is nutrition. Third important aspect in the bacteria is their mode of reproduction. Yes or no? So when it comes to reproduction, bachcho, then bacteria shows only asexual mode of reproduction. Bacteria divide by fission method. There is no true sexual reproduction taking place in bacteria. Bacteria only divide by fission method, which is an asexual method. In bacteria, there is no zygote formation, there is no meiosis, there is no alternation of generation. That means there is no switch from haploid to diploid and diploid to haploid. There is a parent bacteria, it divides, produces daughter bacteria. These daughter bacteria again mature, again divide, new daughter bacteria. Simply fission, simply asexual reproduction, no confusion, no complexity, simple, fast reproduction. Clear? Let's do it. So reproduction, bacteria reproduces asexually by fission method. Where there is a parent bacteria having not so well defined genetic material. Invaginations occurs in the form of mesosomes. They grow, finally reach to the center, genetic material duplicates, and ultimately the bacteria divides into two equal sized daughter cells receiving the same amount of genetic material. Right? This is fission. Now, coming back. However, bacteria do not show true sexual reproduction. True sexual reproduction means what? True sexual reproduction means alternation of generation. As there is no alternation of generation, there is no zygote formation, there is no meiosis, there is no change from haploid to diploid or diploid to haploid. Thus, they do not show any true sexual reproduction. Let's write. No true sexual reproduction means no alternation of generation. That means there is no such kind of activity. That means there is no zygote formation or meiosis. Therefore, we can say bacteria reproduces very quickly because asexual method of reproduction is very simple, fast and quick process. Am I clear with this? Will you all remember all the three functional aspects of bacteria? Respiration, nutrition and reproduction. Respiration, 4 types. Tick. Nutrition, 3 categories. Reproduction, only asexual, no true sexual. No true sexual means no alternation of generation. No alternation of generation means no switch between haploid, diploid, diploid, haploid, as they lack zygote formation and meiosis. Right? Thumbs up for everybody. Yes or no? Very good. Very, very good. Impressive. Proceeding further. So this is the general study. Now we are going to study the organisms of this kingdom, which are further divided into 2 new kingdoms, Archaebacteria and Eubacteria. So first I am going to teach you about Archaebacteria and then we will proceed further to Eubacteria. Archaebacteria, you know, they were separated from the kingdom Monera by Carl Woese on the basis of certain features. Those features were 16S rRNA gene sequencing, structure of plasma membrane, cell wall composition and the nature of genetic material because they contained histone proteins. Correct? Now, Archaebacteria beta, they are one of the primitive most organisms on this earth. These organisms can survive in any extreme environmental conditions. Let's write this point. Very, very important. Go and check the paper of 2012-13. You will find questions on this. Right? So, Archaebacteria are the group of organisms who can survive under extreme environmental conditions. They can survive under extreme environmental conditions. Why? Question is why they can survive. So we have 2 reasons for this. Number 1, I'm writing here as space constraint is there. Their cell wall nature. And number 2 is their cell membrane. You remember we discussed that the cell membrane is made up of lipid monolayer. So this lipid monolayer does not allow the external environmental condition to impact the internal environmental conditions. Number 1. Second, cell wall is made up of pseudomurine. Yes? Yes or no? Yes. So now let me tell you what is pseudomurine. Focus. Pseudomurine beta. First, to understand what is pseudomurine, you should also know what is murine. So murine is actually the type of chemical found in the cell wall of bacteria, which is made up of proteins and sugar. There are 4 different types of amino acids making peptidoglycan and 2 different types of complex sugars, NAG and NAM, N-acetyl muramic acid and N-acetyl glucosamine. However, in Archaebacteria, this NAM is replaced by NAT. NAT stands for N-acetyl talosamine muronic acid. Thus, due to this replacement only, NAM replaced by NAT, Due to this one change only, the composition of cell wall in archaebacteria is known as pseudomurine, and this pseudomurine again make this archaebacteria resistant towards the external environmental conditions. Clear? Shall I write it? Yes, ma'am, you should write it. Okay, so if I want to write it, I should have a space. Okay. So space constraint is there. I will write somewhere here. So cell wall structure, if I talk about, the cell wall structure is pseudomurine, where NAM is replaced by NAT. N-acetylmuramic acid is replaced by N-acetyltalosamine-muronic acid. This gives cell wall a resistance nature. Clear? Now, archaebacteria, my dear students, can be divided into three categories as methanogens, halophils and thermoacidophils. All the three types of archaebacteria can survive in extreme environmental conditions. As if I talk about methanogens, then first feature on the basis of which we are going to classify them is their habit and habitat. Bachcho, methanogens, as the name suggests, they are found in the regions rich in methane because they themselves are methane producers. Correct? Methanogens. Gens means producer. Methano is methane. Therefore, they can be found in the gut of ruminants, in rice fields, in marshy areas. Right? Methanogens. These are methane producers. Thus, found in methane rich regions like gut. You know what is gut? Yes. Gut of ruminants. Ruminants means cows and buffaloes. Okay. Moving to halophiles. Halo means salt. So these are those bacteria which can survive in even extreme salinity. Salinity, which is like in ocean water, they can be found where more than 35% salinity is there. Some halophiles are also found in hypersaline lagoons where salinity may reach up to 100%. So in that extreme saline water, these bacteria can easily survive. Their osmolarity is matched up with the ocean water. Right? If I throw you in the ocean water, you will not be able to survive due to exosmosis. Our osmolarity does not match with the ocean water. But halophiles, their osmolarity is matching with the osmolarity of the ocean water. Hence, they can survive in that extreme environmental conditions as well. So, halophiles are those who can survive in extreme saline conditions. They can survive in extreme saline conditions where salinity is more than 35%, sometimes even 100%. Right? Coming to the third type, thermoacidophils. Name itself suggests. Thermo means temperature, acido means low pH. So, these are those archaebacteria who can survive in extreme high temperature of up to 80 to 100 degrees Celsius and pH as less than 2. Right? So, these bacteria can survive in high temperature, which is about 80 to 100 degree, and low pH, like maybe 2 or even less than 2. Right? So these are the three types of archaebacteria which are found in extreme environmental conditions due to their cell wall nature and plasma membrane nature. Moving ahead, next is on the terms of nutrition. Bachcho, methanogens, they are chemoautotrophs. They can prepare their own food by utilizing chemical as the source of energy. Halophiles, purely heterotrophs, cannot prepare. Thermoacidophils are also chemoautotrophs. Next, mode of respiration. Methanogens, strict anaerobes, obligate anaerobes. Remaining two, they are facultative anaerobes, means normally require oxygen, but even if not given, they can respire anaerobically. So they are facultative anaerobes. Clear? Yes or no? So these are the three types of archaebacteria and their classification system which you should know and remember. Whatsoever question will be asked in NEET will be only from this section. If you remember this much, if you see this video, learn this from the video, read the NCERT, I guarantee you that all the questions will be easily solved. Right? Proceeding further. Organisms of Monera. Organisms of Monera. Now we are going to talk about those bacteria which are true bacteria. And there are different groups in true bacteria, out of which two we are going to discuss as Mycoplasma and Cyanobacteria. Few information we are going to discuss about Mycoplasma and few information we are going to discuss about Cyanobacteria. Are you all ready, guys? Absolutely. Yes, ma'am. Ma'am is also ready. So why to waste time? Start with Mycoplasma. Mycoplasma is the smallest bacteria known, with an average size of 0.3 microns. It is also known as PPLO, pleuropneumonia-like organism. Correct? Now, Mycoplasma do not have cell wall. It lacks cell wall. Hence, do not have a well-defined shape. Right? चलो लिखते हैं इस चीज को. Mycoplasma. Number 1. Smallest bacteria, smallest sized organism. Number 2. Also known as PPLO. Number 3. Lacks cell wall, therefore no defined shape. No defined shape means showing pleomorphy. So they show pleomorphism. Right? Because they lack cell wall, therefore they are also known as bacteria with their coats off. Coats means clothes. They do not have cell wall now. So that is why mycoplasmas are also known as bacteria with their coats off. So also known as bacteria with their coats off. Right? So these are certain points which you should know for mycoplasma. Coming to the next true bacteria and coming to the next organism of Monera, eubacteria, cyanobacteria. Very, very important. This you need to do in little bit of detail. Cyanobacteria, also first of all known as blue-green algae. They are the only bacteria to show proper oxygenic photosynthesis. Photolithoautotrophs, they prepare food, they show photosynthesis, but during photosynthesis, they do not release oxygen as a byproduct. However, cyanobacteria performs oxygenic photosynthesis. Performs oxygenic photosynthesis as they have pigments similar to higher plants. They have chlorophyll A. This is a unique feature in bacteria. Otherwise, normally photolithoautotrophs, they trap light with the help of other pigments like bacteriochlorophyll, but they do not have chlorophyll A. Presence of chlorophyll A is only reported in one type of bacteria, that is cyanobacteria. So they possess chlorophyll A for trapping light. Apart from chlorophyll A, cyanobacteria also have some additional pigments called phycobilins. So they also have some additional pigments called phycobilins. Right? Now, they have proper cell wall, unlike that of mycoplasma. Cell wall is present in cyanobacteria, and that cell wall is 4-layered. So cyanobacteria possess multiple layered cell wall, 4-layered cell wall. Cyanobacteria, bachcho, they are purely aquatic. They are non-motile. They are free floating. Right? I hope all these features which I have written so far, you are able to understand that cyanobacteria, commonly known as blue-green algae, they perform oxygenic photosynthesis as they have chlorophyll A for trapping light. Along with chlorophyll A, they have some additional pigments called phycobilins. Then they are having proper 4-layered cell wall, followed by their aquatic in nature. They lack flagella, hence they are non-motile, plus they simply float on the surface of water. They cannot swim because they do not have flagella. So cyanobacteria, they simply float on the surface of water, hence they are free floating. All these points clear? So we are going to now start the next kingdom after Monera is Protista. You know that the term Protista, this kingdom Protista, was discovered by Ernst Haeckel. This kingdom was coined by Ernst Haeckel. Yes or no? We did it. This fellow, Ernst Haeckel, coined the term Protista, and he separated all the unicellular eukaryotic organisms which were initially placed either in kingdom Plantae or in kingdom Animalia. They separated from there and kept in this new kingdom Protista. So all the members of kingdom Protista are unicellular eukaryotic in nature. Clear? Right. Now, you are coming after, you're studying kingdom Protista, bachcho, after kingdom Monera. And in kingdom Monera, you have seen that organisms are made up of prokaryotic cell which lacks well-defined nucleus, which does not have membrane-bound organelles in its cytoplasm. But now, we are starting with those kingdoms which are made up of eukaryotic cells. Thus, in Protista onwards or from Protista onwards, the cells will possess well-defined genetic material. Well-defined genetic material means the genetic material which is present in the nucleoplasm, as the genetic material is separated from the nuclear membrane. It is separated with the help of nuclear membrane. Plus, the genetic material have histones for packaging. Yes or no? So we can say that in eukaryotes, the genetic material is true nucleus type. Right? The second point, it was discovered by Ernst Haeckel. Right? Now, they possess well-defined nucleus. Well-defined nucleus means when the genetic material is surrounded by nuclear membrane. Is surrounded by nuclear membrane and possess histones for packaging. Now, cytoplasm shows compartmentalization from kingdom Protista onwards due to the presence of membrane-bound cell organelles like ER, Golgi body, etc., mitochondria, etc. They also have ribosomes for protein synthesis, but now the cytoplasmic ribosomes found in Protista are ATS type. So you can mention over here, they possess ATS type of cytoplasmic ribosomes. In Monera, you have seen that the ribosome nature was 70S, but now here the nature of ribosome changes from 70 to 80S. Clear? Yes, very good. Now, majority of the members of kingdom Protista beta, they're aquatic in nature. They may be freshwater or they may be marine, but majority of them are aquatic. So you should know this. Question may come on this line that Protistians, the member of kingdom Protista, are majorly found where? So your answer should be they are majorly aquatic in nature. In aquatic, they may be marine or they may be freshwater. Right? Very good. Now, bacho, since kingdom Protista is made up of eukaryotic cells, and there is one more peculiar feature of eukaryotic cell, that is presence of protein called tubulin, which is responsible for providing cytoskeleton structure of the cell, as well as the cytoplasm of eukaryotic cells also shows cytoplasmic streaming called cyclosis. No more cytoplasm is constant, rather it shows circular movement called cyclosis. So you should know that members of kingdom Protista, their cytoplasm shows movement called cyclosis. Clear? Next point. Now, coming to the very important aspect of any organism of anybody's life is reproduction. So when I talk about reproduction in Protista, then Protista can reproduce by both the methods, sexually as well as asexually. Asexually, they will reproduce maybe by fission method or fragmentation method. But when it comes to sexual reproduction, then Protista shows proper sexual reproduction involving gamete formation, fusion to form zygote. There is a switch between haploid and diploid generations, which is called as proper alternation of generation. So you have to remember that members of kingdom Protista, they show both the type of reproduction, that is asexual and sexual. Sexual reproduction means what? Involving both gamete formation and zygote formation. When gametes and zygotes are formed, this lead to alternation of generation between haploid and diploid stages. From haploid to diploid after fusion, and again from diploid to haploid after meiosis. So this is called as alternation of generation, and it is a peculiar feature of sexual reproduction. Clear? Yes or no? Very good. Now moving to the next point, next general feature of this kingdom Protista. Bacho, the members of kingdom Protista, the boundaries of kingdom Protista, we say, are not well defined. The boundaries of this kingdom are not well defined. Why? Because the organisms which are kept in kingdom Protista, they may be moved to other kingdoms as well. If I talk about Chlamydomonas, if I talk about Chlorella, still their position is in doubtful that whether they should be kept in Protista, as they can also be kept in kingdom Plantae. No doubt we keep them in kingdom Protista only, but yes, some may say that they can be also kept in kingdom Plantae, right? So some organisms of kingdom Protista which are kept in kingdom Protista can also be moved to other kingdoms. Hence, we can say that the boundaries of this kingdom are not properly marketed. Right? Read it. Boundaries of this kingdom are not well defined, as organisms which belong to Protista are more relevant to other groups other than Protista. Clear? Yes or no? For example, Chlamydomonas and Chlorella. Okay, so these are certain general features related to kingdom Protista. Now we are going to study the classification of Protista. Protista, like that of Monera, also shows diversity in terms of nutrition. And on the basis of nutrition itself, we can divide kingdom Protista into three broad groups. That is, producer Protistians, consumer Protistians, and protozoans. In producer Protistians, we have kept those Protistians who can prepare their own food. And they are diatoms, dinoflagellates, and euglenoids. In consumer Protistian, we have kept those Protistians who cannot prepare their own food and feed on dead and decaying organic waste. And under this category, we have kept slime molds. Clear? Last, protozoans, bachcho. It is that category of Protistians which are considered as primitive relative of animals because they do not prepare their own food. Either the members are parasitic or they are saprophytic, or in short, they are depending upon some other sources of food. They cannot prepare their own food. And under protozoan, we have four subcategories as flagellated protozoans, ciliated protozoans, amoeboid protozoans, and sporozoans. One by one, all this classification we have to do in detail. Without wasting time, my dear students, let's start first with producer Protistian. And in producer Protistians, first we are going to talk about diatoms. So are you all ready for diatoms? Yes, very good. Moving ahead. So they belong to a group called chrysophytes, diatoms and desmids. Together they belong to a group called chrysophyta. So first we are going to study chrysophyta under kingdom Protista, which includes diatoms and desmids. This group includes diatoms and desmids. These are producer Protistian. That means these organisms can prepare their own food. They are autotrophic in nature. Now, if they are autotrophic in nature, that means they can trap light and prepare their food. For trapping light, bachcho, they need photosynthetic pigments. That means the members of chrysophyta have photosynthetic pigments, because of which they can trap light and produce food. Now you should know what are those photosynthetic pigments. So the photosynthetic pigments which are found in chrysophytes are, I'm writing it over here, chlorophyll A, chlorophyll C, xanthophyll. They have a special type of xanthophyll called fucoxanthin. This fucoxanthin, my dear students, is actually a brown colored, a golden brown colored pigment. Right? So this fucoxanthin, this presence of fucoxanthin in the members of Chrysophyta impart them slight golden brown color. Hence, they are also known as golden brown algae. Clear? So, diatoms and desmids, they are photosynthetic protistians. They belong to Chrysophyta. They have photosynthetic pigments like chlorophyll A, chlorophyll C, xanthophylls. One special type of xanthophyle found in them is fucoxanthin, which is brown in color. Hence, these organisms may also appear brown in color. Clear? Very good. Now, my dear students, if they can prepare the food, then someday will come when the food will become in excess. Yes or no? So, will they throw their excess food out of the body or will they store it? Yes, they are going to store. When your papa brings excess amount of money and then you utilize the amount which you require, remaining do you throw? No, you keep it in your bank for further use. You reserve your extra money. Similarly, these organisms can prepare their food, but when it becomes excess, they store it in the form of leucocene oil, also called as chrysolaminarin. Clear? Let's write this point. The reserve food material in them is leucocene. In bracket, you can write chrysolaminarin. Right? Clear? Now, question comes, ma'am, where are they present? So, as I told you, majority of the protistians, they are aquatic. So, diatoms and desmids, they are also aquatic, and majorly they are free floating organisms found on the surface of water. They are free floating, that means they lack flagella. They are non-motile organisms. See, so now they are microscopic because they are unicellular, you know that. Float passively, they are free floating. Why? Why do they float passively in water current? Because they lack flagella. Clear? Next point, told you, they are photosynthetic in nature. They can prepare their own food. Now, you know why can they prepare their own food? They have their own pigments. Excess food is also getting stored. Moving ahead towards their body structure. Now, if I talk about the body organization of diatoms, then it has been observed that the body organization of diatoms is like biscuit box. It is like tin box. You know, biscuit box or tin box, which is having two halves. There's one upper half and there is one lower half. And always remember, if you want to close that box, then the diameter of the lid should be more than the diameter of the dabba. Yes or no? The box, the lid diameter should be more than the diameter of your actual box. That means the upper half diameter is always more than the lower half, so that both fits properly into each other. Similarly, in diatoms also, body is in diatoms. Body organization, body is similar to soap box like or tin box like or biscuit box like, soap box like. How? As it is divided into two halves. There is one upper half which is known as epitheca. What do we call upper half as? You call upper half as epitheca. And then there is a lower half called hypotheca. And remember, the diameter of epitheca is going to be more than hypotheca, so that the body fits into each other well. I hope you have understood this. Now I am going to show you with the help of a simple diagram. Okay, so let me remove some points from here. Okay, right now I'll just draw it over here. Yes, see. So this is how the body of diatom is. Can you see this? It is like a box. It's like a soap box, tin box, biscuit box, having how many halves? 2 halves. This is the upper half, which is called as epitheca, and this is the lower half, which is called as hypotheca. Can you see the upper half? Epitheca is having more diameter as compared to lower half, that is hypotheca. Correct? Now, inside this, it encloses the fluid, which is cytoplasm. It's all cytoplasm. There's a large central vacuole inside which is present the nucleus. Right, and all other cell organelles are also present in the cytoplasm along with ATS ribosome, because it is represented by eukaryotic cell. Yes or no? I hope the structure of diatom is clear to each and every one of you. Now, next and very important point from the point of view of NEET, bachcho, the cell wall is present in diatoms. The cell wall of diatom is rich in silica. Silica is an indestructible chemical. Diatoms may die, but their cell wall, due to their indestructible cell wall, they will not undergo decomposition. Now, for example, this is my ocean. They're chiefly found in ocean water. They are also called as chief producers in ocean. Now, they are free floating diatoms. So diatoms are floating over here. They will die. Once they will die, their body will stop floating and will get deposited at the seabed. They are going to form heaps of diatoms on the seabed. Now they cannot be decomposed because of the presence of indestructible chemical called silica in their cell wall. As a result, diatoms keep on depositing on the seabed, forming heaps of diatoms. And these heaps of diatoms is known as diatomaceous earth or Kieselguhr. Right? So these are diatomaceous earth. This diatomaceous earth is of great economic importance. Why? It is slightly gritty in appearance and it can be used in making syrups, toothpaste, nail paints, in fact varnishes, the paints which you use in your home. So all these uses diatomaceous earth. Yes or no? Let me repeat once again. Due to the presence of their walls are embedded with silica. Silica is an indestructible chemical. As a result, the wall of diatoms also become indestructible. Once they die, they settle down at the seabed, forms their heaps, they pile up. They are called as, thus diatoms have left behind large amount of cell wall deposits in their habitat. Accumulation over billions of years referred to as diatomaceous earth. Yes or no? Very good. Now moving ahead. Now these diatomaceous earth, they are gritty in appearance and they are having so many economical benefits. Like for example, they can be used, as I told you, in nail paints it can be used, in varnishes it can be used in making toothpaste, your syrups and etc. Clear? Now, one more important point. Diatoms are also called as chief producers in the ocean. Diatoms are simply free floating organisms on the surface of water. So they are maximally exposed to light. They are on the surface now. They are maximally exposed to light. Hence they trap as much as amount of light possible and perform maximum photosynthesis. Therefore known as chief producers in the ocean. Clear? Yes or no? So these are the points which you have to remember for diatoms under the heading Chrysophyta. I hope this thing is clear to all my dear students. One more time I am going to show you the body organization, the structure of diatoms, and then we will move to dinoflagellates. That is the second producer protisteans. So how is the body? Let's revise and then let's proceed further. How is the body? The body is soap box like, represented by two halves. The upper half, which is known as epitheca, and the lower half, which is known as hypotheca. The diameter of epitheca is more than the diameter of hypotheca, so that they both fit into each other well. Clear? Inside is the cytoplasm, large central vacuole in which there's a nucleus present. Apart from this, they have all the membrane bound organelles here and there, all ribosomes and etc. Clear? They are non-motile, they do not have flagella. That is why they are free floating. Right? Now let's talk about their reproduction also. Little bit I'm going to tell you about their reproduction. Normally, Protista can reproduce by both sexual as well as asexual method. However, when the conditions are favorable, these protisteans prefer what? Asexual method of reproduction. So diatoms under favorable conditions undergo fission. Remember this point? Write it. Diatoms reproduces asexually by fission method, reproduces asexually by fission method under favorable conditions. However, when the condition becomes unfavorable, then diatoms switches their mode from asexual to sexual. Clear? Right. However, when the condition becomes unfavorable, the mode of reproduction changes from asexual to sexual. So just remember this thing, that diatoms prefer asexual mode of reproduction over sexual. Clear? Now moving ahead towards the next group of protista, producer protista, dinoflagellates. Dinoflagellates are included under the group Pyrophyta. Clear? So they are included under the group Pyrophyta. Like diatoms and desmids are included under Chrysophyta, so dinoflagellates are included under Pyrophyta. Please remember, not given in NCERT but important for you. Chalo. So now let's start discussing about the dinoflagellates. Dinoflagellates, beta, they are also unicellular, eukaryotic, photosynthetic protisteans. Dinoflagellates, similar to that of diatoms, they can also photosynthesize and they can also prepare their own food. So they are photosynthetic protistians. Again, why? What is the reason that they can also prepare their own food? Same reason, presence of photosynthetic pigments. So, my dear lovely students, dinoflagellates, first of all, they're aquatic. They prefer to live in marine water. So, they are mostly marine and they are photosynthetic in nature. Why photosynthetic? Due to the presence of photosynthetic pigments like chlorophyll A, chlorophyll C, xanthophylls. They may also have different type of xanthophylls. Some may have fucoxanthin, some may have another type of xanthophylls like peridinine. So they may have xanthophylls like fucoxanthin or peridinine. My dear students, fucoxanthin is a brown colored pigment. So dinoflagellates having fucoxanthin in high amount will also appear to be golden brown in color. And peridinine is the it is a pigment, it's a photosynthetic pigment which is red in color. So a dinoflagellate having peridinine will appear in which color? Red in color. Remember this thing? Very good. Now, you know they have photosynthetic pigment. They can prepare their own food. So, if they can prepare their own food, a day will come, excess food is happening. Will they throw out? No. They are also going to store. And dinoflagellates, my dear students, store food in the form of carbohydrates or oils. Carbohydrate or oils. Clear? Very good. Now, unlike that of diatoms, dinoflagellates are motile organisms. They do not float, rather they swim on the water surface. Why do they swim? Because they have flagella with them. They are flagellated dinoflagellates. And how many flagellas do they have? They have 2 flagella placed perpendicular to each other. I'll show you with the help of a diagram. Clear? So now I'm going to show you a dinoflagellate, a simple representation of a unicellular photosynthetic protistian over here. Try to understand, try to see. So yes, this I'm just trying to draw a simple dinoflagellate over here. Some diagrams are given in your NCERT as well. Now you can see I'm drawing 2 openings. Yes or no? Yes, very good. Right? So this is how dinoflagellate outer appearance will be. Now, their cell plate is represented is actually stiff cellulosic cell plate is present in them, which is called as theca or lorica. So the cell wall which is in dinoflagellates is stiff in nature due to the presence of cellulosic plates, and such stiff cellulosic plates are known as theca or lorica. So let me write the name. So this is how these are the stiff cellulosic plates, right? And due to the presence of, my dear students, these stiff cellulosic plates It seems to be like as if dinoflagellate is wearing an armor. It has protected its body from the outside conditions. The stiff cellulosic plates gives an appearance of what? Armor around dinoflagellates. Therefore, dinoflagellates are also known as armored organism. You know what is armor? Armor is the one which old time knights, the soldiers used to wear. All those made up of metals and all, na, so that they can be protected from the swords and other weapons of the enemy. So similarly, dinoflagellates, it seems they're wearing an armor of stiff cellulosic plates to protect them from the outside environmental conditions. Right? So this is how the dinoflagellate will appear. Now you can say two grooves. Can you see two grooves? One is transverse and another is longitudinal. So this is one groove and this is another groove. And in each groove are placed one flagella. I told you these are flagellated organisms. How many flagella? Two. Both the flagellas are placed perpendicular to each other, each in one groove. One in longitudinal groove, another in transverse groove. So what are these? These are the flagella, the locomotory organ. Beta, they undergo whirling movement. Flagella in its position is going to swirl, right? And it is going to help in the movement of dinoflagellate. Such movement is called as whirling movement because the flagellas are swirling in their position. And this is helping in the movement of dinoflagellate. Clear? Very good. Moving ahead. So most of them have two flagellas. One is in the longitudinal groove and second is in the transverse groove or furrow. Clear? Between the wall plates. You can see it very clearly in the diagram. Now, moving ahead. So I hope all these points are clear. The structure of dinoflagellate, flagella, whirling movement. Let me write it over here. Dinoflagellates shows which type of movement? They show whirling movement. Clear? Now, moving ahead, coming to the point peridinine. You remember I told you that dinoflagellates may also have a red colored pigment called peridinine. So the dinoflagellates which are having peridinine, they will appear to be of which color? They will appear to be red in color. So next point is related to that. There are some dinoflagellates, some red colored dinoflagellates like Goniolax and Gymnodinium, which are responsible for causing red tides. How? They rapidly reproduce by asexual method. They undergo fission. They increase their number on the surface of water. And when tides come, then those tides appear red in color due to large number of these red colored dinoflagellate. I am repeating it once again. Pay attention that red dinoflagellates like Goniolax and Gymnodinium, due to their rapid multiplication by fission method, they rapidly increase their number on the ocean water. Ocean water surface is having large number of red colored dinoflagellates. So when tides are going to come, those tides will also appear red in color. And such tides are known as red tides. Right? Very good. So very often, red dinoflagellates like example Goniolax and Gymnodinium is also one of the example, undergo rapid multiplication and that makes the sea appear red. Not sea appear red. Basically, red sea is caused by cyanobacteria Trichodesmium erythraeum. Here you just remember, sea will appear red, but specifically the denotion is with respect to the tides. It is going to cause the red tide. Clear? If the question comes that which organism causes red sea, then your answer should be Cyanobacteria Trichodesmium erythraeum. If question comes, which organism causes red tide, then your answer should be Goniolux and Gymnodinium. Please highlight whatever I have said right now. Pin this video for this line. Very important. NEET is going to ask this question. Clear? Moving ahead. Now, the one example which I have mentioned, Goniolux, apart from causing red tide, it also produces a toxin called saxitoxin. Bachcho, it is a neurotoxin, right? If it is intaken by the fishes, then it causes paralytic shellfish poisoning. It affects the vertebrate organisms. This toxin, this neurotoxin, affects the vertebrate organism and causes PSP in fishes. Let's see. Toxins released by such large numbers. Which large numbers? Goniolix. Goniolux is a red colored dinoflagellate which also produces a toxin called, which toxin? Saxitoxin. Will you remember the name? Yes. It is which kind of toxin? It's a neurotoxin and it affects the functioning of vertebrate organism and it causes paralytic shellfish poisoning. Clear? Very good. In fishes. So it causes death of animals such as fishes. And which is that disease which is caused in those fishes? It is PSP. Paralytic shellfish poisoning. So which is the toxin released by Goniolux? Quickly. Saxitoxin. Which kind of toxin is this? Neurotoxin. It affects vertebrate organism and it may kill some marine organisms like large fishes. And those fishes may die due to PSP. Clear? Okay, so very good. This is all about dinoflagellates. I hope diatoms and dinoflagellates both are clear to all my lovely students. Moving ahead towards the next photosynthetic protistian, that is Euglena. Euglenoids. Euglenoids, the most common example in euglenoid is Euglena viridis. Right? Now, let's try to understand about euglenas. Euglenas, they are richly found in fresh water and that fresh water should be stagnant. So, first and foremost line is that these are the organisms which are found in fresh water, stagnant fresh water. Clear? Number 1 point. Second, if I talk about their body organization, then their body is somewhat like conical in shape. How conical? The upper part is broad and as you go down, the body starts tapering. So it's like an inverted triangle. Upper part is broad. As you go down, the lower part becomes narrow. Euglenas, beta, they lack cell wall and their outermost boundary is represented by proteinaceous covering called pellicle. So instead of cell wall, they have a protein-rich layer called flex pellicle. Now due to cell wall is not there, so constant shape is not maintained. Hence, their body organization is flexible. Clear? Now, shall I draw the diagram of Euglena? How the body is, it's like inverted triangle. See. So this is how Euglena is. You can see the upper part is broad and as we go down, it becomes tapering. This outermost boundary is pellicle. It's a eukaryotic, single-celled eukaryotic organism. So inside it, it will have all membrane-bound cell organelles and nucleus, everything. So it is going to have a big nucleus. In nucleus means genetic material along with the nuclear membrane. Then they have all the cell organelles. Here I am going to show you some chloroplast. Why? Because these are photosynthetic protistians and being photosynthetic protistian, they should have chloroplast in them. So this is chloroplast. Now, my dear lovely students, the photosynthetic pigments found in Euglena are similar to that of higher plants. Their chloroplast have pigments chlorophyll A and chlorophyll B along with xanthophylls. So whatever the pigments are found in Euglena is similar to what? Is similar to higher plants, as they have chlorophyll A and chlorophyll B along with xanthophylls and carotenoids. Right? So next point is, okay, have I written? Yes, the pigments of euglenoids are identical to those present in higher plants. Hence, whatever photosynthesis takes place, it takes place with the help of these pigments. Similarly, beta, they are also photosynthetic. They will also prepare their own food, and a time will come they will try to store it. So if they store it in the form of paramyelin bodies. Clear? Now they are found in water, so they also need some contractile vacuoles for maintaining the osmoregulation. So they also have contractile vacuole for maintaining osmoregulation. Apart from this, my dear students, Euglena are also motile organisms. They are motile due to the presence of flagella. How many flagella? 2 flagella. They are eukaryotic organisms, so their flagella arise from basal bodies. So there are basal bodies, right? First basal bodies are produced, and from those basal body arises flagella. How many flagella? 2. One short and another long. So there are 2 unequal sized heteroconti flagella. One is long flagella that you see outside, and second is short flagella rising from the basal body that soon merges with the longer flagella. Chota wala is going to soon merge with the longer flagella. So there are how many flagellas? 2. A short one and a long one. Soon the shorter one merges with the longer one, and what you see outside is only one flagella. Clear? Yes or no? Thus Euglena also shows flagellar movement. This flagella is going to swirl in order to allow the movement of Euglena. Right? Yes or no? The point of meeting, where the 2 flagellas meet, this point of meeting is known as paraflagellar region. Now, my dear students, flagella has this Euglena has one special property. That is, it shows mixotrophic mode of nutrition. It shows what? It shows mixotrophic mode of nutrition. What is mixotrophic mode of nutrition? That means Euglena can act as both autotrophic and heterotrophic depending upon the conditions provided. For example, if light is present, then these euglenas are going to trap light and prepare their own food. But when they are deprived of light, then they change their mode of nutrition from autotrophic to heterotrophic, and they start predating. They become predators. They start predating on other small organisms. That means their mode of nutrition is mixotrophic. Why? Because in the presence of light, they trap light, prepare their own food. However, if light is not there, in that case, they change their mode of nutrition from auto to hetero. They become predator. They start feeding on other small organisms near to them. I hope euglena is also clear. So these are the points which you have to remember for your NEET examination with respect to all the three photosynthetic protisteans. We are completed with diatoms, dinoflagellates, as well as euglena. So photosynthetic protistans are done. Moving ahead to next group, and that next group is consumer protisteans, that includes slime molds. So far we have completed with the photosynthetic protisteans, which includes diatoms, dinoflagellates and euglenoids. Now we are heading towards the next group of protista on the basis of mode of nutrition, that is slime molds. Slime molds, my dear students, are consumer protists who are not able to prepare their own food. Rather, they depend upon some other organisms. They feed on dead and decaying organic matter. They are saprophytic protisteans, as they feed on dead and decaying organic matter. They feed on, the body moves along decaying twigs and leaves, engulfing the organic material. Clear? If I talk about the body organization of slime molds, then slime molds, the vegetative body of slime mold is represented by a mass of multinucleate cell wall-less structure called plasmodium. So this is the plasmodium, which is the body of slime molds. It is the vegetative body, cell wall-less body of slime mold, which is multinucleate. And it is produced when the conditions are favorable. So during favorable conditions, under suitable conditions, slime molds, they form an aggregation called plasmodium, and this plasmodium size may grow up to feet long. So the main body of slime mold is what? Plasmodium. Now, when conditions become unfavorable, like maybe water availability, light availability, nutrient availability, something is not perfect. In that case, condition will switch from favorable to unfavorable. In that scenario, this plasmodium will produce a fruiting body. This plasmodium will produce a fruiting body bearing a swollen structure on the tip. So this is the fruiting body bearing a swollen structure on the tip. So read it. During unfavorable conditions, plasmodium produces a fruiting body bearing a swollen structure on the tip. Now here division takes place inside this swollen tip to produce large number of spores. So after division, number of spores are produced. A time comes when this structure, when this swollen structure, it ruptures. This swollen structure is going to rupture to release the spores outside. Remember beta, the vegetative body is wall-less. It is not having cell wall, but the spores which are produced after the rupture of the structure on the fruiting body, these spores are resistant, highly resistant, because of the presence of cell wall around them. And now, whenever the favorable conditions comes, first of all, these spores which are released, they are dispersed through air current, and whenever they get a favorable condition, they germinate and they grow into a new slime mold. Clear? So next statement says the spores, they possess true walls. Next, which are extremely resistant and can survive for many years. They are dispersed through air current. Now you can see in slime mold one very common thing. This plasmodium is wall-less, without cell wall. Right? So this cell wall-less feature of plasmodium is similar to animal-like organisms. Fruiting body. Fruiting body is a fungi-like feature, and presence of cell wall around the spore is a plant-like feature. So can we say that slime molds are the connecting link? They are showing some features related to animal, related to fungi, and related to plant. So slime molds are connecting link in Protista, which shows some features similar to animals, similar to plant, and similar to fungi. Hope this small concept of slime mold is clear to everybody, and questions will surely come only from the section which I have told. So focus on them, revise them, learn them. Moving ahead with the last group of Protista, which is protozoans. Protozoans are bachcho considered as primitive relatives of animals. Why? Why are they considered as primitive relative animals? Because their mode of nutrition is heterotrophic. Their mode of nutrition is heterotrophic. They may either be predator or parasitic or saprophytic, but they cannot prepare their own food. Hence, they are considered as the primitive relatives of animals. Protozoans can further be divided into four as amoeboid protozoans, ciliated protozoans, flagellated protozoans, and sporozoans. Clear? So there are how many types of protozoans? Four. Amoeboid, flagellated, ciliated, and sporozoans. Correct? Now let's talk about these different types of protozoans. First feature on the basis of which we are going to study them is their habit and habitat. Amoeboid protozoans, beta, they may be aquatic, may be terrestrial, they may be found on moist soil, they may be parasitic as well. So if I talk about the habit and habitat of amoeboid protozoans, so they may be found growing on land, on moist soil, on wood, as parasites, etc. So amoeboid protozoans, you can write that they grow either on land or water, may be found on moist soil, etc. If I talk about ciliated protozoans, then they are usually aquatic. Some forms may also be terrestrial, but usually they are aquatic. Then mode of nutrition may be predation, may be parasitism. If I talk about flagellated, flagellated also may be aquatic, or some forms may be terrestrial, they may be parasitic, terrestrial. If I talk about sporozoans, then my dear students, sporozoans are exclusively all endoparasites. They all are endoparasites. That means they grow inside the host body. Clear? Because of which they lack locomotory organ. They don't have any locomotory organ. Thus, the second feature on the basis of which we are going to study protozoan is locomotory organ. Amoeboid protozoans, as the name suggests, they have pseudopodia for locomotion. If I talk about ciliated, then they have ciliary locomotion. Flagellated, flagella for locomotion. Sporozoans, being endoparasites, they do not have any locomotory organ. Clear? Next, some special and exclusive feature which is found in respective groups only. In case of amoeboid protozoan students, some marine forms have been reported which have silica surfaces, silica shells. So this become a feature, special feature of amoeboid protozoans. Marine forms may possess silica shells. Coming to ciliated में one very good example is paramecium. It is a ciliated protozoan which is also called a slipper-like animal. Now, in paramecium, you must have observed they have a specific opening for the ingestion of food called gullet. So, ciliated protozoans are those group of protozoans which have a specific opening for ingestion as well as egestion. So, they have distinct openings for egestion and ingestion. They have a special opening called gullet for ingestion of food. For ingestion of food. Talking about flagellated protozoans, beta, they show rare sexual reproduction. And they prefer to live either as commensal symbionts or parasites. So they are rare sexual reproduction and prefers to live as commensals or parasites or symbionts, etc. Special feature in case of sporozoans is that if you talk about sporozoans, one very good example of sporozoan which I can recall is Plasmodium. It causes a very dangerous disease called malaria. In all sporozoans, it has been observed, my dear student, that infectious, if I talk about the life cycle of different sporozoans, then it is observed that throughout their life cycle, some of the other stages do come which are infectious, and infectious stage of the life cycle is actually responsible for causing the disease. So we can write that infectious spore-like stage appears in their life cycle, right? Example for amoeboid, you can remember the example of entamoeba. For ciliated, you can remember paramecium. For flagellated, bachcho, you can remember trypanosoma, which causes African sleeping sickness. And for sporozoans, I have already told you Plasmodium. So with this, your entire Protista kingdom is over. Hope you have understood this well. So far, we have completed Monera kingdom, including Archaebacteria and Protista. So we have completed with kingdom Monera as well as kingdom Protista. Now students, we are jumping into the third kingdom, which is kingdom Fungi. Are you all excited? Not drained out now? Very good. No one should be drained out. It's very, very interesting, very, very important. Keep up your spirits high, keep yourself motivated throughout the lecture. So now we are starting with the third kingdom, which is kingdom Fungi. Bachcho, you know kingdom Fungi was coined by R.H. Whitaker on the basis of the mode of nutrition. R.H. Whitaker observed that members of fungi are kept in kingdom Plantae. Fungi are heterotrophic, they cannot prepare their own food. Then why are they kept in the kingdom Plantae, where all other organisms are photosynthetic? So R.H. Whitaker removed all the members of fungi and established themselves as an independent kingdom. So first you should know this, write this, learn this, that kingdom Fungi was established by R.H. Whitaker, however you pronounce, on the basis of mode of nutrition. Yes or no, guys? Yes. Very good. Now, this is the first and foremost thing which you should know regarding kingdom fungi. Now, proceeding further, now you have to see other general features of this kingdom. First of all, you know that this is a group of eukaryotic, mainly multicellular, heterotrophic organisms. So, first point itself says it's a group of multicellular. Exception may prevail, one or two, but majority of them are multicellular, eukaryotic. Heterotrophic organisms, that means these are those organisms who cannot prepare their own food. They depend upon dead and decaying organic matter. They may be saprophytic in terms of nutrition. Clear? Moving ahead. Second point, cell wall. Fungi, they have cell wall, but their cell wall is not made up of cellulose, rather it is made up of another polysaccharide called chitin, and chitin is chemically, it is a polymer of sugar, NAG. NAG stands for N-acetyl glucosamine. Very important point. So usually fungal cell wall is chitinous. Exception is there, but when I will teach you the classification of fungi, there I will tell you that which fungi, specifically the lowermost fungi, Oomycetes, their cell wall is cellulosic. However, if the question comes in general that the cell wall of fungi is, then your answer should be chitinous. Chitin ki bani hai. What is chitin? Chitin is chemically, it's a polymer of NAG. NAG is N-acetyl glucosamine. Clear to everybody? Very good. Moving ahead. Third point. Majority of them are terrestrial. Yes. Majority of the fungi, they are terrestrial. Few of them, primitive fungi, may be aquatic. Otherwise, these fungi, either they are growing on dung, or they are growing on skin, or they are growing on tree bark, etc. So, majority of the fungi, my dear students, they are terrestrial. Some primitive fungi may be aquatic. Clear? Fungi, my dear students, prefer to grow in warm and humid climate. They grow well in warm and humid climate. That is why Mummy used to keep food in refrigerator. Mummy keeps food in refrigerator. Why? Why does not she keep the food open for throughout the night? Why she keep it in the refrigerator? The only reason is outside the refrigerator, the condition may become warm and humid, and it will lead to the contamination of your food. Fungus may start growing. That just to avoid that thing, Mummy keeps the entire leftover food in the refrigerator where temperature is cool and not humid, rather it is dry. Thus, such conditions will not allow the growth of fungus. So, where fungus grows? Fungus grows well in warm and humid conditions. Clear? Okay. Now, coming back to the first point itself, I have written that they are multicellular. Do you know any example? Do you know any exception? Example of multicellular fungi is everything. Albugo, penicillium, etc. Whatever you can think of. They all are multicellular. But can you give me any one example of exception in multicellular fungi? Yes, very good. Absolutely correct. Exception is yeast. Yeast is a unicellular fungus and it is commonly used in making bread and beer. You know that this yeast can be used in making bread dough. That is why it is known as baker's yeast, and it is also used in making beer. It is also used in brewing industries as ferment because it helps in fermentation. Hence, yeast is also known as brewer's yeast. Here somewhere I have written, yes, yeast is a unicellular fungi which is used in making bread and beer. Because it is used in making bread, hence yeast is also known as baker's yeast. And because it is used in brewing industries for making beer, hence it is also known as brewer's yeast. Will you remember? Very, very important. This line is also going to come in class 12th. So please remember over here and then revise in class 12th. Going back to the original page, I hope all the first three general features is clear related to fungi. Moving ahead. As I told you, fungi comprises of eukaryotic organisms. So eukaryotic means those organisms who have a well-defined genetic material. They have true nucleus in their cytoplasm. Do you all agree? Raise your hand. Yes, correct. And apart from true nucleus, fungi also contains their cytoplasm is compartmentalized due to the presence of membrane bound organelles like Golgi bodies, like endoplasmic reticulum, like mitochondria, but plastids are not found. Why plastids are absent? Because fungi are heterotrophic. They do not need plastids. They cannot perform photosynthesis. So, question will come. Which cell organelle is absent in fungi? Your answer should be plastids. Now, there is one cell organelle called Golgi bodies. You all know structurally, Golgi body is represented by a number of flattened sac-like interconnected structures called cisterna. There are multiple cisternas, yes or no, in one Golgi body. However, in fungi, Golgi body are unicisternal. That means in fungi, Golgi bodies are only represented by one cisterna. They are not packets of cisterna. In fungi, Golgi bodies are unicisternal. Will you remember this? Yes or no? Yes, give me a thumbs up. Very good. So they are unicisternal. Clear? Fourth point. Go through it once again. Read it by your own. Yes. Moving ahead. Now coming to the body structure. If I talk about the body, then the body of fungi is thallus-like. Thallus means undifferentiated body, not having true root, stems or leaves. Rather, their body is made up of group of haploid cells. So if suppose this is a body of fungus. For example, this is a body of fungus. So this body is made up of number of cells now. It's a multicellular body. So these multicell bodies, each cell, the ploidy of each cell is N. Made up of number of cells. And ploidy of each cell is N. And when the body is represented by group of haploid cells, then that body is called thallus-like. Undifferentiated. No true root, stems and leaves. Clear? So what is thallus-like? Body which is made up of haploid cells. Right? And their body is it's thallus-like. Clear? Now when the body is thallus-like, so the life cycle pattern will be haplontic. That means their zygote, whenever they will show sexual reproduction, when the zygote will be produced, that zygote will undergo immediate reduction division. So haplontic life cycle is exhibited by fungi, which is also known as zygotic meiosis, because as soon as zygote will be produced, it will undergo reduction division to again produce haploid group of cells. Clear? So body is thallus-like, life cycle is haplontic. Haplontic also known as zygotic meiosis. Why? Because the zygote, as soon as it is produced, it undergoes reduction division. Proceeding further. Now we are talking about the body of fungi. So let's continue. So the body of fungi is thallus-like and it is represented by thin thread-like structures called hypha. It is represented by thin thread-like structures called hypha. So single unit, the basic unit in the body of fungi is hyphae, which is a thin thread-like filamentous structure. Number of hyphae, they intermingle. Number of hyphae, they intermingle to form a loose tissue kind of body organization called mycelium. So the body of fungi is represented by a network of hyphae, mesh of hyphae called mycelium. Clear or not? Very good. There is one exception. There is one fungi which do not form mycelium. Can you guess? Giving you 5 seconds, 10 seconds. Can you guess a fungi whose body will not be represented by mycelium? And along with the reason. Not only the answer, I want the reason as well. Yes, very good. The answer is on the board itself. Yeast. Yeast, my dear student, it is a unicellular organism. Being a unicellular organism, how will it produce hyphae? Hyphae itself is a multi-cell structure. So yeast is the only one, it's a unicellular fungi whose body is not made up of hyphae or mycelium. Then what body organization will be called for yeast? It will be called as pseudo-hyphae. Okay? Hyphae term we can use because it is a member of fungi, but not true hyphae, so pseudo-hyphae. Or you can simply say it is a unicellular fungi. If you want to call it in terms of hyphae, then pseudo-hyphae. Am I clear or not? Let's write it. So body is represented by thin thread-like hyphae, and number of hyphae intermingles to form mycelium. Exception: yeast. Why? Because it is unicellular. Am I clear? Very good. Now, talking about the body of fungi itself, let's talk about hyphae. Now, these hyphae in detail. Hyphae, my dear students, may be septate or aseptate. Clear? Now, what is septate? What is aseptate? Ma'am, what all of this? Chalo, let's see. So, I'm drawing two hyphae. In some of the fungi, rather if I say in higher fungi, in advanced fungi, the hyphae, the mycelium is made up of septate. Septates means, septa means cross walls, divisions, partitions. So, such hyphae which are partitioned with the help of walls, then such hyphae are known as septate hyphae. However, in case of lower or primitive fungi, the hyphae in their body, they are not septate. It is continuous like a hollow tube. So such hyphae where no partitions, no cross walls are present, they are known as aseptate hyphae. Presence of aseptate hyphae is a feature of lower or primitive fungi. Correct? Whereas presence of septate hyphae is a feature of higher or advanced fungi. Now, these septate hyphae may be either uninucleate, like this, that means in each segment there is one nucleus, or they may be binucleate. Clear? They may uni or binucleate depending upon the condition. I will tell you that condition. But aseptate hyphae, they are cenocytic, they are multinucleate, because all the nucleus will appear together. They're not separated by walls now. So this aseptate hyphae seems to be cenocytic, multinucleate. Clear? And this septate hyphae can be either uni or binucleate. Yes or no? So we can say that hyphae can be either aseptate, that means no partitions. As a result, condition becomes multinucleate, observed in lower fungi. Can be septate, that means partitions are found. Acha, one more thing, my dear students. Bacha, you can see I have made the partitions, but I have made discontinuous partitions. Can you see? See, some openings are there. See, openings are there now in middle. So remember, partitions are never continuous. They are discontinuous. Some openings are there. Why? Just to maintain the continuity of cytoplasmic flow within the hyphae. Correct? So just to make the continuity of the flow of cytoplasm. So partitions are there, but they are discontinuous to allow the flow of cytoplasmic material. Remember this thing. Now, these septate hyphae may be either monokaryotic, means uninucleate, or can be dikaryotic, that means binucleate. So yes, they can be either monokaryotic or dikaryotic. Dikaryotic means binucleate. Di means two, karyo means nucleus. Monokaryon, mono means one, karyo means nucleus. Monokaryotic septate hyphae means vegetative hyphae, the hyphae which has still not undergone reproduction, whereas binucleate or dikaryotic hyphae represents reproductive hyphae. Why? Because the exchange of genetic material has took place. The nucleus, can you see? Exchange of genetic material has took place in between each segment of the hyphae. This marks reproduction. So hyphae having dikaryotic condition is reproductive hyphae. Hyphae having uninucleate condition represents vegetative hyphae. So monokaryotic means you can also call it as vegetative hyphae, and dikaryotic you can also call it as reproductive hyphae. Yes or no? Clear? So what I have told you, let me sum up for you very quickly. So the body of fungi is thallus-like, undifferentiated. It is represented by thin thread-like, pipe-like structures called hyphae. Hyphae, they mesh, they form network-like structure called mycelium. In case of lower fungi, the hyphae are continuous, then there are no partitions, hence the condition becomes multinucleate. But in advanced fungi, in higher fungi, these hyphae, they are having partitions, septas. However, those septas are discontinuous for maintaining the flow of cytoplasm. Now, since they are septate, condition may be monokaryotic or dikaryotic. Monokaryotic represents vegetative hyphae and dikaryotic represents reproductive hyphae, as it has undergone reproduction. Exchange of genetic material has taken place. Clear? Very good. Now, moving ahead. So this is how the body of fungi appears to be. Hyphae and all. I hope all the features which I have written so far is clear to each and every student. moving ahead towards the next property of fungi, very, very important, that is reproduction. So fungi being a lower, so not well developed kind of organism, can reproduce by all the three methods, that is vegetative, sexual and asexual. Vegetatively it reproduces by fragmentation method. Asexually also fungi can reproduce by producing different types of spores, and sexually it also reproduces by forming gametes and zygote. Here we are more concerned. Fragmentation is very simple. When a part of a fungi detaches from the body, gets the favorable condition, can develop into a new organism. So this type of reproduction is vegetative, is fragmentation. Let's talk about asexual and sexual reproduction. First, I'm going to tell you about asexual reproduction, that how fungi reproduces asexually by sporulation method, and then we will discuss sexual reproduction as well. Clear? Shall we move ahead? Yes, very good. So first of all, we are going to talk about asexual reproduction in fungi, which is done with the help of sporulation method. Fungi produces different types of spores asexually. Such spores are called as mitospores. Why? Because these spores are produced after mitosis. Clear? That is why these spores are known as mitospores. Remember this point. Asexual spores can also be known as mitospores. Why? Because these spores are produced after mitosis. Now there are different groups of fungi. Each group produces its own kind of asexual spore. One by one, I am going to tell you about all those types of spores. First is zoospore. My dear students, zoospores, zoo itself means something related to motility. So zoospores are the thin-walled motile spores produced in lower fungi like oomycetes during favorable conditions. So zoospores are thin-walled spores. Motile means planospore. Motility is plano. Non-motility is aplano. So they are motile spores, means planospores produced during favorable condition. Observed in case of lower fungi like oomycetes. Right? Now, this motility is due to the presence of flagella. Now, zoospore may have either one flagella, right? Or they may be motile due to the presence of two flagella. So, motile are zoospores. They may be due to one flagella, or may be two flagellas may be present. If there are two flagellas, then both the flagellas may be present at the head of the zoospore, and if two flagellas not present on the head, then they may present laterally as well. Once again, I am repeating, the first asexual spore which now I am telling you, which is observed in lowermost group of fungi oomycetes, is zoospores. Zoospores are thin-walled mitospores, planospores produced during favorable conditions. They are plano due to the presence of flagella. Number of flagella may be 1 or 2. If there are two flagella, then both the flagellas may be present at the head of the zoospore, or they may be present laterally. Clear? These spores, beta, they are produced within a covering. First of all, suppose this is a fungi, right? This fungi is going to produce a stalk-like structure. On that stalk-like structure, a swollen tip is going to appear, inside which these zoospores will be produced. The tip is going to burst, releasing the motile zoospores, and whenever they will get the favorable condition, they will start germinating into a new fungi. Because these spores are produced within a covering, that is why they are known as endogenous spores. Endo matlab andar, genus matlab banna. So these are endogenous spores. Why? Because they are produced within a covering. Clear? Moving ahead towards the next asexual or mitospore, which is, this AC is, my god, it's killing me. The temperature is too low. Okay, so moving ahead, sporangiospores. Sporangiospores are basically, they are also asexual or mitospores, which are observed in case of another group of lower fungi, zygomycetes. So first of all, they are also thin-walled, produced during favorable conditions. Third, they are non-motile. They are aplanospores. They are also endogenous spores, my dear students. They are also produced within a covering. And next, they are observed in case of zygomycetes. Clear? Moving ahead, third type of asexual spore is conidia. Conidia are also asexual spores which are observed in case of higher fungi like ascomycetes and deuteromycetes. So first of all, conidia, let me draw conidia over here. Yes. Conidia. These are also asexual mitospores. They are also non-motile spores. Aplanospores. They are also thin-walled. Why? Because they are produced during favorable conditions. Observed in case of higher groups of fungi like ascomycetes and deuteromycetes. But they are exogenously produced. Unlike that of zoospores and sporangiospores, conidia are exogenously produced on the stalk called conidiophore. I'll show you how. So there is stalk which is produced in case of ascomycetes and deuteromycetes called conidiophore. Right? On these stalks are present some bottle-shaped structures, and on these bottle-shaped structures comes in chains spores called conidia. So can you see? These are the chains of spores called conidia, which are borne on the stalk called conidiophore. Are they covered within any covering? क्या इनके अराउंड कोई covering आपको दिख रही है? No. That means they are produced exposed. They are not produced within a covering. Such spores are exogenously produced. So remember this thing, conidia are the asexual spores produced in case of asco and deuteromycetes during favorable conditions exogenously. They are also non-motile, borne on a stalk called conidiophore. Clear? So these are the list of asexual spores. There is one more asexual spore called chlamydospores. Chlamydospores are the only thick-walled spores, non-motile spores, which are produced during unfavorable conditions. The three which I told you so far, zoospore, sporangiospores and conidia, all three are produced during favorable condition, hence they are thin-walled. But if I talk about chlamydospores, then chlamydospores, my dear students, they are thick-walled spores produced during unfavorable condition in case of fungi. Right? So this much you have to do for asexual reproduction in fungi. What you have to remember, fungi can reproduce asexually by sporulation method. It produces different types of spores depending upon the group of fungi. Can be zoospore, can be conidia, can be sporangiospores or chlamydospores. One note point. Basidiomycetes is the only group of fungi that do not reproduce asexually. It only reproduces vegetatively by fragmentation method. What have I said? Have you heard it properly? Shall I repeat? Listen to me. Basidiomycetes is the only group of fungi which does not produces asexually. It only shows either sexual reproduction or vegetative reproduction through fragmentation. Correct? Okay, very nice. Proceeding further, now let's discuss about the sexual reproduction in fungi. So fungi can also reproduce sexually by the method of formation of gamete and their fusion to form zygote. So if I talk about sexual reproduction, so first I will divide this into two, because in lower fungi like oomycetes and zygomycetes there is one specific method of sexual reproduction going on, and in higher fungi like asco and basidio, remember deuteromycetes do not exhibit sexual reproduction. ठीक? So in case of lower fungi, lower fungi includes oomycetes and zygomycetes, whereas higher fungi to show sexual reproduction includes ascomycetes and basidiomycetes, not deutero. Why? Because deuteromycetes, the members of deuteromycetes, their sexual reproduction is yet not reported. Now, in case of lower fungi, that is oomycetes and zygomycetes, sexual reproduction is very easy. It is done in 3 steps. First is plasmogamy. Second is karyogamy. And third is meiosis. What is this? Plasmogamy means fusion of the protoplasm. So suppose if I talk about oomycetes, they have produced their spores. Now spores, first of all, they will come in contact. Suppose these are the 2 sexual spores of oomycetes or zygomycetes. They are 2 gametes. Spores means gametes over here. Meiospores, or you can say gametes. So 2 gametes of oo or zygomycetes have come in contact. So what will happen? Initially, their protoplasm is going to fuse. That fusion of their protoplasm is known as plasmogamy. As soon as fusion of their protoplasm takes place, instantly their 2 nuclei, their 2 independent nuclei will also fuse. Fusion of nuclei is known as karyogamy. Correct? Now, as soon as this fusion of the 2 nuclei takes place, Soon after plasmogamy and karyogamy, simultaneously a diploid zygote will be produced. And this diploid zygote, I told you, life cycle of fungi is haplontic, zygotic meiosis. So this zygote will instantly undergo meiosis to release some haploid spores. So these are the gametes. Gamete 1, gamete 2, these are the meiospores. Right? So this 3-stage simple sexual reproduction is observed in case of lower fungi, plasmogamy, directly karyogamy, and then meiosis to produce haploid spores. But what happens in case of higher fungi, that is Asco and Basidio? There is one intermediate stage between plasmogamy and karyogamy. What happens? Their gametes, their gametangia, when they come in contact, plasmogamy takes place, but sometimes initially, instantly, karyogamy does not take place. The two gametes, they will fuse. What happens? Plasmogamy takes place, fusion of protoplasm takes place, but as seen in case of lower fungi, that in lower fungi both plasmogamy and karyogamy is taking place simultaneously. However, in higher fungi, it is not taking place simultaneously. There is a dikaryotic stage. There is a N plus N condition appears called dikaryotic stage. Di means two nuclei. So instant after plasmogamy, due to no nuclear fusion, a partial stage comes called dikaryotic stage. This dikaryotic stage is short-lived in case of Ascomycetes, whereas it is long-lived in case of Basidiomycetes. Now after dikaryotic stage, then comes your karyogamy, where the fusion of the nucleus takes place to produce a diploid zygote, and now this zygote will undergo meiosis to produce haploid meiospores. Correct? So what is the difference between sexual reproduction of lower fungi and higher fungi? Lower fungi reproduction is done in 3 steps: plasmo, karyo, meiosis. But in case of higher fungi, there is one more additional step in between plasmogamy and karyogamy by the name dikaryotic stage. It is actually a partial N plus N. It's a partial N plus N condition that arises temporarily. It is short-lived in case of Asco and long-lived in case of Basidio. Then comes karyogamy, zygote is produced, and then that zygote undergoes meiosis to yield haploid spores. Clear? So this is how sexual reproduction takes place in case of fungi. Now after meiosis, meiospores are released. You have to remember some names of the meiospores produced in different groups of fungi. Let's discuss them. So sexual spores or meiospores: oospore, ascospore, basidiospore and zygospores. Oospores, beta, as the name suggests, these are the meiospores which are produced in group of fungi Oomycetes. Oomycetes, lower fungi, gametes formed, plasmogamy, karyogamy, zygote, meiosis, spores. Spores will be called as oospores. So oospores are the spores produced. These are meiospores in Oomycetes. Correct? Next, ascospores. Ascospores name suggests these are the meiospores produced in Ascomycetes. You have to remember some more things in case of Asco and Basidiospores. Beta, ascospores are endogenously produced spores. Suppose this is a fungi. Fungi forms a fruiting body. Ascomycetes forms a fruiting body called ascocarp with a swollen tip called ascus. Inside the ascus, gametes fuse to form zygote, and now this zygote will undergo reduction division. Inside this ascus, reduction division takes place to produce haploid meiospores, which are called as ascospores. So since ascospores are produced within a covering, these are endogenous spores. Clear? Observed in ascomycetes. Coming to third type of sexual spore, which is observed in case of basidiomycetes. Remember, basidiomycetes reproduces sexually by forming basidiospores. My dear students, basidiospores are exogenously produced. How? Suppose this is a basidiomycetes. It also produces a fruiting body. Tick. This fruiting body is called as basidiocarp. Now, this basidiocarp on the tip will have a club-shaped structure like this, not a spherical, not like this, not like a sac-like structure, not, rather it will have a club-shaped structure called basidium. On these basidium will be some sterigmatas, some bottle-shaped structures called sterigmata, where these sexual spores will be produced in chains. So these are basidiospores. Correct? So what are basidiospores? Basidiospores are exogenously produced spores on the basidium, on the fruiting body basidiocarp in basidiomycetes. These are meiospores because they are produced after meiosis. They are produced after reduction division. Clear? So will you remember these points? Yes, thumbs up. Very good. Last is zygospores. As the name suggests, these are produced in group of zygomycetes. So remember this thing. These are produced in zygomycetes. Clear? So these are the names, these are the list of meiospores produced in different groups of fungi. Lower fungi, sexual reproduction 3 stages. Higher fungi, sexual reproduction in 4 stages. I hope this sexual reproduction, asexual, everything, all the general features related to fungi is clear in each and every student. Now moving ahead to the next topic and very important topic of fungi, that is classification. I am using these terms oomycetes, zygomycetes, asco, basidio. Have I told you? No. Now I am going to tell you that why am I using all these terms. Come. So scientists studying fungi actually divided fungi into broad 5 groups on the basis of morphology of their mycelium, whether this hyphae is septate or aseptate, then mode of spore formation, which type of meiospore, which type of mitospore, type of fruiting body if they are having, and etc. So all on those features, the fungi is divided into some groups. Those groups are Phycomycetes, which is of lower fungi. It includes oomycetes and zygomycetes. They both are lower fungi. Followed by ascomycetes, basidiomycetes and deuteromycetes. Deuteromycetes, beta, it is just an additional group of fungi where all those fungi, higher fungi are kept, whose nature of sexual reproduction is still unknown. But if their sexual life cycle is discovered, then the members from Deuteromycetes are majorly either moved to Ascomycetes, and sometimes they're also moved to Basidiomycetes. Majorly to Ascomycetes because the other features of Deuteromycetes are very much similar to Ascomycetes. So what I have said, I'm writing it here in the note points. The members of Deuteromycetes are those whose members of Deuteromycetes are those whose sexual life cycle is yet not decoded. But as soon as it gets decoded, the member of Deuteromycetes is then moved. Once it is discovered, then the member of Deuteromycetes is moved either to Asco or to Basidio, but majorly to Asco. Why? Because members of Deuteromycetes, they show more similarity with Ascomycetes. Clear? Yes or no? Chalo. So these are the 5 classifications of fungi on the basis of morphology of mycelium, mode of spore formation, fruiting body, habit and habitat, etc. Now I am going to teach you all these 5 groups of classification in a very simple manner with the help of a chart. Right? My dear students, if you remember this chart, then all your problems will be solved. No trouble will come to you. So please try to remember the chart which I am drawing, which I am making it right now. All the points related to the classification of fungi will come on your tips. So let's start. Yes or no? Come on. So the first feature on the basis of which we are going to divide them is on the basis of their habit and habitat. Except Oomycetes, all other fungi groups are terrestrial. Oomycetes, my dear students, they are aquatic. Otherwise, they are all terrestrial. ठीक. Second feature, cell wall. Cell wall. I told you na, cell wall is chitinous, but there is one exception. Oomycetes, my dear, their cell wall is cellulosic, and in all remaining it is chitinous. Yes or no? Tell me. Clear? Shall I move ahead? Very good. Next point. Third, mode of nature of mycelium, structure of mycelium. In case of lower fungi, you remember mycelium is, hyphae is aseptate, unbranched and cenocytic. So in दोनों के लिए you can write hyphae is aseptate, unbranched and multinucleate. Any doubts? We have done in the starting of fungi classification. In all these 3 higher fungi, hyphae is septate and branched. Now, in case of Asco and Basidio, my dear, they may be either uninucleate or binucleate if the members of Ascomycetes and Basidiomycetes have yet not undergone sexual reproduction, then their hyphae will be uninucleate. Otherwise, they will be binucleate. So, the hyphae septate, branched, and either monokaryotic or dikaryotic. But what in case of Deuteromycetes? Deuteromycetes hyphae are septate and branched, but will they ever become dikaryotic? No. Why? Because Deuteromycetes do not show sexual reproduction. There will be no exchange of genetic material. Thus, in case of Deuteromycetes, their hyphae will be septate, branched, and monokaryotic. Yes or no? Clear? Very good. Moving ahead. Nature of spore. There are two types of spore, myo and mito. So first we are going to discuss about their mitospores, asexual spores. You know everything we have done. You just have to mention what you have learned previously into this chart. So mitospores, oomycetes, zoospores. Zygomycetes, sporangiospores. Ascomycetes, exogenously produced. Conidia. Basidiomycetes? Yes, come on. Very good. Basidiomycetes do not show asexual reproduction, so no spores, but reproduces vegetatively by fragmentation method. And Deuteromycetes, similar to Ascomycetes, reproduces asexually by Conidia. Clear? Next, meiospores. Oomycetes, oospores. Zygomycetes, zygospores. Ascomycetes, ascospores, born on fruiting body called ascocarp. Basidiomycetes, basidiospores. Deuteromycetes, no sexual reproduction, no meiospores. Correct? Yes or no? Can you remember this whole chart? Yes, can you correlate with whatever I have taught previously in this chart? Yes or no? Yes, very good. Now last is examples which you have to learn. Examples, my dear students, only learn from NCERT, no here and there examples. So in case of oomycetes, you can remember examples like albugo candida, which causes white rust in crucifers. Very good. Apart from albugo, you can also remember example like saprolignia. Correct? Yes or no? Very good. Now, moving ahead. Zygomycetes. Example, bread mold, rhizopus, mucor mucido, which grows on cow dung. It's a coprophilous fungus. Ascomycetes. So many examples. One or two I will mention over here. Remaining you can learn from your NCERT. Yeast, penicillium, neurospora, claviceps. Neurospora is called as Drosophila of plant kingdom because it is used extensively in genetic engineering. Claviceps causes ergotism. It causes ergot in rye. Penicillium, you know, first antibiotic. Yeast, unicellular fungi, which is used in making bread and beer. Basidiomycetes. Example, the edible one, mushroom. Some disease causing fungi are like Puccinia, which causes rust disease, Astilago, which causes smut disease. Correct? Deuteromycetes, Alternaria solani, which causes early blight of potato. Apart from Alternaria solani, then you have Helminthosporium, then Trichoderma, and so on. So all these examples you can directly learn from the NCERT, and please focus on the notes plus NCERT. That's it for the classification of fungi. Hope it is clear to each and every student. Whole fungi kingdom is finally over. I hope you all enjoyed till here. Now all the major kingdoms which we have to cover in this chapter are done. The last topics of the chapter which are left are viruses, mycorrhiza and lichens. So which we are going to do now. Okay, so I hope fungi is clear to everybody. Now before starting with viruses, let me give you some more important information regarding the classification of fungi. Right now I have explained you the general features of kingdom fungi, that how they reproduce, what is their organization, body structure as well as classification. But now I am just going to give you 2 to 3 some extra information regarding the various classification of fungi, that is also going to be equivalently important from the point of view of NEET. So as you can see on your board, I have written the different classification scheme of fungi on the basis of their morphology of mycelium, on the basis of their sporulation, type of fruiting body, etc. You have also seen the chart of classification. Now, according to this, Phycomycetes, which can be further divided into 2, that is the lower fungi, Oomycetes and Zygomycetes, followed by the higher fungi that includes Ascomycetes, Basidiomycetes and Deuteromycetes. Now bachcho, the lower fungi Oomycetes is also known as algal fungi. Why Oomycetes? The other name, the general name for Oomycetes is algal fungi, and it is known as algal fungi because of 2 reasons. First of all, they are usually aquatic in nature, and second, their cell wall is cellulosic. You know that in fungi, cell wall is chitinous, but in the members of Oomycetes, it has been seen that their cell wall is cellulosic in nature. Moving ahead with Zygomycetes, Zygomycetes can also be known as conjugation fungi. Moving ahead with Ascomycetes. Ascomycetes, you have seen that their fruiting body was ascocarp, which bear a sac-like ascus on the top, where ascospores are produced. So because of that sac-like ascus, this Ascomycetes is also known as sac fungi. On similar grounds, Basidiomycetes is also known as club fungi because the fruiting body basidiocarp bears a club-shaped structure called basidium. So in relation to that club-shaped basidium, Basidiomycetes are also known as club fungi. Moving ahead, Deuteromycetes, my dear students, they are also known as fungi imperfecti. And the reason for them being called as fungi imperfecti is that they do not show true sexual reproduction. The members of Deuteromycetes, in their life cycle, sexual reproduction is yet not reported. That is why they are called as fungi imperfecti. These additional name of each group of fungi should be known to you. Questions may be asked from this section as well. So now your fungi group is done. Moving ahead, the next two kingdoms are kingdom Plantae and kingdom Animalia. Kingdom Plantae, you know, we are going to do it as a next chapter, and kingdom Animalia also you study in detail in your zoology. So all the 5 kingdoms, 3 kingdoms I have covered so far. 4th kingdom I am going to cover in the next chapter. 5th kingdom is done in your zoology. So overall, the kingdom classification, the kingdom details is done. But there are some organisms like viruses, subviral agents, mycorrhiza, lichens. These are also considered as living organisms but could not find a suitable place in any of the 5 kingdom classification by R.H. Whitaker. So now I am going to teach you all those organisms who could not find any position, any suitable position for themselves in the 5 kingdom classification of Whitaker. Along with the reasons, I am also going to tell you the reasons that why such inhuman thing has been done to these organisms and they could not find a suitable place in their classification system. So let's start with next topic, viruses and subviral agents. So yes, moving ahead with next and very important topic, viruses and subviral agent. As I told you, these are the organisms who did not find a suitable place in Whitaker's 5 kingdom classification. Why? The question comes, why? The reason to this question is viruses, my dear student, they are acellular organisms. These organisms, they are not even made up of one cell. They are actually exception to cell theory. Now, Whitaker classification include all those organisms who follow cell theory, whose body is at least made up of one cell or group of cells. But when he saw viruses, then he observed one thing which was very uncommon, that viruses are not even made up of one cell. So viruses are acellular organism. They do not follow cell theory. Hence, they could not find a suitable place in Whitaker's 5 kingdom classification. Apart from them being acellular, there are many more features which are associated with viruses, because of which again they could not find a suitable place for themselves. Viruses are actually, my dear students, connecting link between living and non-living. They show certain features similar to living organisms and certain features similar to non-living. Therefore, Whitaker could not understand that whether viruses are living or not. If they are not living, then surely they cannot be a part of 5 kingdom classification. And if they are living, then again they are acellular. According to cell theory, all living organisms are made up of cells, and viruses, if we consider them as living, then why their body is not even made up of one cell? So there was a lot of confusion in the mind of Whitaker. Hence, he excluded viruses and the subviral agents from his kingdom classification. Clear? Very good. See, so viruses are acellular organisms and they are connecting link between living and non-living. I will explain you how are they connecting link. But before that, the term virus was coined by Pasteur. The term virus means venom or poison. So the term virus was coined by Pasteur, which means venom or poison. They are acellular organism. They are the connecting link between living and non-living. Why are they connecting link? So what are those features that make them similar to living, and what are those features which make them similar to non-living? Let's discuss. So first I'm going to share all those features of viruses similar to living organisms. Number 1, presence of genetic material. Viruses have genetic material either in the form of DNA or RNA, but never both. And presence of genetic material is a living property. No chair, table, fan, lights, AC will have genetic material. Yes or no? Because they are non-living. And anything which is living will definitely have genetic material. So, in viruses, presence of genetic material was reported. So, write this point. The first property of them being similar to living object is presence of genetic material, which can be either DNA or RNA, but never both. Remember this thing. If in place of or I write and, then the statement becomes wrong. Clear? Second feature, because of which they are considered as living, they can be considered as living. They undergo mutation. Their genetic material may show mutation, and mutation is a feature of living objects. So second reason, they undergo mutation. Number third reason, some viruses have also been observed with enzymes, like for example lysozyme, right? Reverse transcriptase enzyme. So presence of enzyme has also been reported in viruses, which is again a living feature. Write it. That is presence of enzymes. For example, like lysozyme. Clear? Fourth, they can also undergo division. They can multiply. So multiplication, increasing their number, is again a living feature. Your table, your chair, your fan, can they reproduce? Can they increase their number by their own? No. It would have been so good if they would have also been shown reproduction. Bought one AC, overnight it multiplied and made 2 ACs. Bought one fan, it multiplied overnight, made 4 fans. It would have been so good now, but unfortunately non-living objects do not reproduce. But viruses, it has been observed, they can multiply and they can increase their number. So thus they are one of the feature of living organism, can multiply. Next, beta, viruses are thought to be living only when they are present inside the host body. Outside the host body, they are inert. But when they are present inside the host body, then they show living properties. Thus, we can say that viruses are obligate intracellular parasite. So remember this thing, their living property is only when these viruses are present inside the host body. So you can write that viruses are compulsorily obligate intracellular parasites. Clear? I hope all the features are clear to each and every one. Now these are certain features because of which you can consider them as living. But now why do we consider them as non-living? Let's see. So now I'm going to show you all those features because of which viruses is also considered as non-living. And number one reason is they are acellular. Their body is not even made up of one cell. They are thus exception to cell theory. Right? Second, outside the host body, my dear students, viruses can be crystallized. Their crystals have been reported. Their body can be crystallized and can be stored in bottles. Scientist Stanley, I'll tell you, Stanley scientist, he crystallized the crystals of virus TMV, tobacco mosaic virus. So this property of viruses getting converted into crystals is a non-living property now. Our living bodies are not like puzzles. You cannot dismantle my body and again rejoin to make my body like original one. No, it is not possible because I am a living object. You cannot dismantle my body and reorganize it. However, in viruses, their body are inert outside the host body and can be crystallized. So this is again a non-living property. Write it. Second point, their body crystalline, they can be crystallined. And the crystals can be stored inside the bottle wherever you want to keep. Next, in viruses, no energy storing mechanism, no respiratory mechanism has been reported. Now imagine a living body which is without respiratory system, which is without energy storing system. Can you imagine any body without energy storing system, without respiratory system? No. But viruses also do not have such property. Hence, they are considered as non-living. So what? Next feature of viruses being non-living are they lack energy storing system. Next, they also lack respiratory system. And all these properties make them similar to non-living. Now you can see that these are the properties that are making viruses similar to non-living, and these are the properties which are making them similar to living. So what to decide? Therefore, scientists called viruses as connecting link between living and non-living objects. Clear? Yes or no? And this is the reason why viruses did not find any place in Whittaker's 5 kingdom classification. Now, moving ahead, let's discuss the contribution of various scientists in the field of virology. Virology is basically the study of virus. So let me write it over here. Study of virus is known as virology. Clear? Now, moving ahead. First, talking about Pasteur. I've already told you who is Pasteur. He's the one who coined the term virus. So, in front of Pasteur, we can write that he coined the term virus. Now, second scientist Stanley. I already used this name Stanley also. Stanley is the one who for the first time crystallized the virus TMV and told that the crystals are largely made up of proteins. Clear? So who is Stanley? Stanley is the one who crystallized TMV. He crystallized TMV. TMV is tobacco mosaic virus. So he crystallized TMV and said that the crystals are largely made up of proteins. Clear? Moving ahead. Third scientist is Ivanovsky. Ivanovsky was the scientist who said that he was studying on TMV, tobacco mosaic virus, and said that viruses are even smaller than bacteria because these viruses can easily pass through the filter paper of bacteria. So Ivanovsky was the scientist who explained about the size of the virus. So he was studying on TMV. While studying TMV, he told that viruses, these particles, the ones which he was studying, TMV, virus, the ones which was studying, viruses are even smaller than bacteria. Why? Because they can easily pass through bacterial filter paper. Correct? Last is scientist Bejring. Bejring was the scientist who said he actually proposed a statement for virus as contagium vivum fluidum. He gave a very cool statement for viruses as contagium vivum fluidum, means infectious living fluid. According to him, according to this fellow, according to Bejring, if an organism who's infected with virus, if the fluid from that organism is infected to another healthy person, then that healthy person also develops the same symptoms as it was present in an unhealthy body. So what he said, Bejring, I'm repeating it once again. For viruses, he stated a statement, contagium vivum fluidum, which means infectious living fluid. First of all, why he proposed this statement for virus? Because in his experiment, he found that when a fluid from infectious person, a body who is infected with virus, so if the fluid from that body is injected and given to a healthy person, then that healthy person will also develop the same symptoms as observed in unhealthy body. So healthy body will also become unhealthy. Why? Because the extract of the unhealthy body was transferred in the body of healthy person. So viruses actually got transferred and gave the symptoms in the healthy body as well. That means they are infectious in nature. They are infectious living fluid. Clear? So what Bejring did? This fellow, he infected or he injected a fluid from unhealthy to healthy body and observed the same symptoms in it. Right? That is why he proposed the statement contagium vivum fluidum. Now, moving ahead, types of viruses. Now, before starting with the types of viruses, you should be aware about the structure of viruses. Now, you know that viruses are not even made up of one cell. Then what is the body of virus made up of? So your answer will be that viruses are nucleoproteinaceous structures. Their body is made up of nucleic acid and protein. Structurally, virus can have three parts: envelope, capsid, which is their body, and body enclosing the genetic material. So before starting with types of virus, here I'll just use the space. So before starting with types of virus, first let me tell you about the structure of virus. Structurally, virus, my dear students, consists of three parts. Envelope may or may not be present. It is not compulsory. Second is capsid. Capsid is actually the body of the virus, and this capsid is made up of proteins. And the body that is capsid, inside is present the genetic material, which can be either DNA or RNA. Right? Envelope is actually made up of proteins, which is contributed by the virus, which is contributed by the capsid, plus lipids and carbohydrates that come from the host body. And presence of envelope is not a compulsory factor. Envelope may be found in viruses like HIV or herpes, but it is not a compulsory factor. So structurally, virus is made up of three parts: envelope, capsid. Like for example, this is a capsid. This is a capsid which is made up of proteins. Okay, and each unit of capsid is known as capsomere. So this is capsid, and each unit of capsid is known as capsomere. And this capsid or capsomeres are actually proteinaceous. And inside it, they enclose the genetic material, which can be either DNA or RNA. Now, around this capsid, envelope may or may not be present. Right? If envelope is present, then it is made up of protein contributed by the virus, lipids and carbohydrate contributed by the host. Clear? May be present, may not be present. So this is how a simple virus will look like. So therefore you can say that viruses are nucleoproteinaceous structure. Why? Because their main body is made up of protein that encloses the nucleic acid, that is your genetic material. Now moving ahead with the types of viruses. So in your course there are two viruses which you have to study. One is tobacco mosaic virus and second is bacteriophage virus. First, let's start with TMV. As the name suggests, it is a virus that infects the tobacco plant. Correct? So what is TMV? It is a virus that infects tobacco plant. Structurally, it consists of capsid and encloses the genetic material. Capsid, which is made up of capsomeres. Total number of capsomeres recorded in a TMV is 2130. So structurally, TMV is made up of capsid and genetic material. Capsid is obviously the body, the proteinaceous body, and the basic unit of capsid is capsomere. How many capsomeres are found in TMV? Near about 2130 capsomeres. Whereas if I talk about the genetic material, then do remember in TMV the genetic material is single-stranded RNA. No DNA is found in TMV, and RNA, whether it is single-stranded, yes. So genetic material in case of TMV is single-stranded RNA. Single-stranded RNA is made up of 6,400 nucleotides. So, capsomeres are 2,130, whereas number of nucleotides in RNA is 6,400. Clear? So, yes, now moving ahead. Okay, if I talk about the size of TMV, then approximately the length and the breadth, if I talk about this, the length and the diameter of TMV is about 3000 angstrom is the length, and the diameter is about 180 angstrom. So this is the standard size of TMV. Clear? Yes or no? So this is how you can draw the body of a TMV. Suppose this is the body that is capsid, which is made up of number of capsomeres. Correct? How many capsomeres? 2,130. Apart from this, then enclose the genetic material. The genetic material, which is single-stranded RNA having 6,400 nucleotides. Clear? And their length and diameter is about 3,000 and 180, 3,000 angstrom and 180 angstrom, length and diameter. Correct? So this much you have to do for TMV. Moving ahead towards the next set of viruses, which are bacteriophage. My dear students, bacteriophage are the viruses. The name itself suggests the virus which attack bacteria. Bacteria killing viruses are bacteriophages. Right? Now, structurally, their body is represented by a polygonal head, a neck region and a tail region. So first I'm going to draw the bacteriophage and then we are going to study. So what is bacteriophage? It is a virus that attacks bacteria. Bacteria killing virus. Now I'm going to draw the structure. It is having a distinct polygonal head. Right? This head is made up of, or this head contains enzymes like lysozyme, as well as they have genetic material which is double-stranded DNA. So they have genetic material which is double-stranded DNA. Very important question comes from this section. TMV has single-stranded RNA, whereas bacteriophage has double-stranded DNA. Where it is present? It is present in their polygonal head region. Right? Now, head and neck region are connected with the help of collar. Wait. Okay. So now, there's a short collar. Followed by a neck region. And then there is a long tail. So this is a long tail which ends with 6 tail plates. 3 on one side and 3 on another side. So these are 6 tail plates. This is how the bacteriophage appears to be. There's a head, collar, a small collar, just like your body. There's a head, there's a collar, neck, tail region, and then there are 6 tail plates, 3 on one side, 3 on another. These tail plates, my dear student, they help bacteriophage to get attached on the surface of bacteria. So tail plates are responsible for helping in the attachment of bacteriophage on the substratum, which is usually the bacteria. The head also contains enzyme like lysozyme. Right? So this is how bacteriophage appears to be. Question will be asked directly on their genetic material and role of tail plates. And what is bacteriophage? I hope types of viruses is also clear. This much you have to do for viruses. What are we supposed to learn for viruses? First of all, why are they considered as connecting link? Why did not they get any position in Whittaker's 5 kingdom classification? Third, types of viruses, structure of viruses, correct? And contribution of different scientists. Now moving ahead towards the end part of the chapter by discussing the subviral agents. There are 3 subviral agents reported. These are those viral agents which are even smaller than viruses. We say that viruses are even smaller than bacteria. Yes or no? You have seen in the contribution of scientists that viruses are those which can even pass through the filter paper of bacteria. That means viruses are even smaller than bacteria. Now some scientists also discovered particles which are even more smaller than viruses. And there are 3: viroids, virusoids and prions. Little bit information you should know regarding all the 3. Number 1 is viroids. So now we are starting with subviral agents. Number 1 subviral agent which we are going to discuss is viroids. Viroids, my dear students, they were discovered by Theo Diner. This scientist name you have to remember. That is why I am telling you. First, second point. My dear students, viroids are only, the body of viroids are only represented by the genetic material. They do not have any capsid like that of viruses. In viruses, the body, the structure of viruses was represented by capsid and genetic material, correct? But in viroids, Diner reported the presence of low molecular weight RNA, which is not protected by any proteinaceous capsid. So what are viroids? Viroids are represented by low molecular weight RNA but lacks protein coat around themselves. These are subviral infectious agents. Infectious agents how? Because they can cause disease like for example PSTD, potato spindle tuber disease. They are infectious in case of plants. Right? Okay. So they were discovered by Theo Diner. Second point, they are represented by low molecular weight RNA as genetic material. Third, lacks capsid, lacks protein coat. They are infectious and cause disease like for example PSTD, which stands for potato spindle tuber disease, chrysanthemum disease called chrysanthemum stunt, citrus exocortis. So there are some disease which are caused by viroids that you should remember. PSTD stands for potato spindle tuber disease. Apart from this, they can also cause chrysanthemum stunt disease. Chrysanthemum stunt disease. Clear? So this is the little information regarding viroids which you should know. I am repeating it once again. Viroids discovered by Diner. They are represented by low molecular weight RNA that lacks protein coat around itself. They are infectious in nature and cause disease like PSTD or chrysanthemum stunt. Right? Now, the next subviral agents are virusoids. Virusoids are even smaller than viruses, you know. They are subviral agent and they do not have any independent existence. These virusoids are the subviral agent which are reported inside the body of some other larger viruses. Correct? So what are virusoids? Virusoids are the subviral agents that are present inside some other larger virus. Present inside some other larger virus. Now can you see? Viruses themselves are so small and virusoids are even smaller than viruses that for their existence they need a shelter of some other larger virus. So these are virusoids. Third comes prions. Prions are infectious proteins. They are subviral agent and their structure is structurally prions are only protein. They lack genetic material. Clear? So you can say that prions are those subviral agents whose body is represented by infectious proteins. Correct? These are simply infectious proteinaceous structures and they can cause number of disease. So write about prions. Prions are subviral agents which are actually highly infectious in nature, represented by only proteins. And cause number of disease like for example bovine spongiform encephalopathy. Second, Kuru disease. Third, scrappy sheep disease, etc. Kuru disease is also known as laughing death disease. So, bovine spongiform encephalopathy, Kuru disease also known as laughing death disease, scrappy sheep disease. These are the names of different diseases which have been reported due to prions, and structurally prions are infectious proteinaceous particles. They are subviral agents as they are even smaller than virus. So there are three subviral agents: viroids, virusoids and prions. I hope all these three are clear. Now my dear students, apart from viruses, even mycorrhiza and lichens are also two such organisms which could not find any place in Whittaker's classification. However, there's a different reason to it, unlike that of viruses. My dear students, mycorrhiza and lichens are actually composite structures of two different organisms. If I talk about, first I'll talk about lichens and then I'll come to mycorrhiza. So first if I talk about lichens, lichen is a composite structure which is made up of two different organisms, algae and fungi. Now algae are the member of Plantae and fungi itself is a new kingdom. So Whittaker got confused that where to keep lichens, whether in kingdom Plantae or whether in kingdom fungi. So he thought not to keep them into any kingdom, let's keep them independently. So lichens are living organisms, but as their composite structure of two different organisms belonging to two different kingdoms, hence could not find a place in Whittaker's classification system. Clear? So what are lichens? Let's discuss. Lichens, first of all, they are the composite structures, composite unit of two organisms Basically, algae and fungi, they are mutually associated. Lichen is a mutual association of fungi and algae, where fungi and algae both are getting equal benefit. And that is why they have decided to live together as one unit. Clear? Chalo, so let's write in that manner. So lichens are the mutual association between algae and fungi, right? The algal partner in lichen Yes, the algal partner in lichen is known as phycobiont. Why? Because the study of algae is phycology, whereas fungal partner is known as mycobiont. Why? Because the study of fungi is known as mycology. Clear? Repeating once again, pay attention. Lichens are the mutual association between algae and fungi. Mutual means the two different organisms have decided to live together as they are equally getting benefited. The algal partner is known as phycobiont and the fungal partner is known as mycobiont. This algal partner is basically the member of chlorophyceae, sometimes cyanophyceae also. So majorly chlorophyceae. Sometimes cyanophyceae. Sometimes a bacteria, cyanobacteria, may also get associated with fungi to form lichens. But usually it is the green algae which gets associated with fungi to form lichens. Moving ahead, fungal partner, majorly the fungi which contribute in lichen formation are the members of Ascomycetes. But sometimes some members of Basidiomycetes may also contribute in the formation of lichens. Okay? Now, third point, this is the first point. Second point, now the third point. In lichen, 95% of the lichen body is made up of fungi and 5% of the body is only of algae. So I have to remember the major partner. Out of the two organisms, the major partner is fungi and the minor partner is algae. So fungi is contributing also a lot. I'll tell you the functions of each component in lichen, and with the contribution only you can decide that fungi is a dominant partner as compared to algae. So who is the dominant partner in case of lichens? The dominant partner is fungi. Why fungi is dominant partner? Now I am going to tell you. Why fungi is a dominant partner, students? Because fungi is contributing a lot in making of lichens. Now see, fungi knows how to absorb water and mineral. Fungi know how to reproduce. Fungi know how to make anchorage, means attachment to the substratum, but does not know how to prepare food. Right? What am I saying? Listen to me again. Fungi is like a father. Okay, leave everything. Now listen. Fungi is like a father in a home. See, what is the role of the father? Father goes to the office, does the work, brings money. With that money, you can buy the food items, you can make a home and everything. Father can bring the raw material with the help of the money, But who is going to use those raw material in order to cook food? So that is the role of the mother. Correct? So in lichen also, fungi is the father and algae is the mother. How? Fungi goes to the market, brings the raw material for the food preparation, that is water and mineral. Second, it also makes the home, it makes the complete anchorage of the organism. Third, it also help in the reproduction, but does not know how to use those raw material for preparing food. Hence, algae is required now. Algae comes in the picture, utilizes the water and mineral absorbed by the fungi in order to prepare food. So the role of algae is limited in preparation of the food. This is the benefit which fungi is getting. In return, algae is getting the shelter, it is getting the raw material for the preparation. Clear? So therefore, both these partners have decided to stay together as a composite unit. Now let's write. So what function is algae doing? Functions of algae. Algae simply performing photosynthesis by utilizing the raw material provided by fungi. Now, if the algal partner is the member of Cyanophyceae, then apart from photosynthesis, it can also perform nitrogen fixation. That is an additional role, and it only happens when the algal partner is the member of Cyanophyceae. But here I am keeping simple and crisp. I am considering that this algae is a member of Chlorophyceae and it is only responsible for performing photosynthesis and preparing food. Now, so this is the role and function of algae. Now, role of fungi in lichen. Why it is a major partner? Why it is dominating? Because number one, it collects the raw material. It absorbs water and mineral for photosynthesis. Second, it makes the shelter. It prepares the anchorage for the lichen on the substratum. Third, it also helps in reproduction. So you yourself can see that the function performed by fungi as compared to algae is more. Hence, fungi is a dominant partner in lichen. But still, dominant partner doesn't mean if father is dominating in the home, that doesn't mean that your home can run without your mother. If mother goes out even for one day, the whole home gets disturbed. Yes or no? So mother is also equivalently important. Algae is equivalently important in lichen. If algae escapes, automatically fungi will die due to lack of food. And if fungi escapes, automatically algae will die in absence of water and mineral. So that is why it is a mutual association. Clear? One more last point. My dear students, lichens are indicators of pollution. They are good indicators of air pollution. Why? Because lichens do not prefer to grow in sulfur dioxide rich medium. So remember this thing, lichens are air pollution indicator. Why? Because they do not prefer to grow in SO2 rich medium. Do not grow in SO2 rich medium. Clear? Apart from this, lichens have many important roles. Some lichens are edible, some lichens are used in making perfumes, medicines, etc. But what you have to remember is simple. They are pollution indicators. what is fungi, what is lichen made up of, what are the role of its each partner, that's it. Clear? Now, apart from lichens, there are one more such organism who could not find a place in Whittaker classification, and that is mycorrhiza. As lichen, mycorrhiza is also a mutual association between two different living organisms, fungi and higher plants. Plants, planty kingdom, fungi, fungi kingdom. Whittaker got confused where to keep it, so he decided to leave them as it is. Hence, mycorrhiza also did not find a place in the classification system. So what is mycorrhiza? Mycorrhiza is also a mutualistic, same, association between fungi and roots of higher plants. Some plants like pinus. Now what happens in some plants like pinus? Their roots are woolly. Their roots do not spread deep in the soil. Their roots lack root cap, right? So they are not able to trap water and mineral with high efficiency. Now if the plant is not going to, if the roots are not going to absorb water and mineral, what the leaves will do? How will they perform photosynthesis? And like this, the plant will die. So plants like pinus, they have a big problem. They are confused what to do. We are having roots, but they are of no use because their roots are woolly. They do not spread deep in the soil. They lack root cap and they cannot perform absorption of water and mineral efficiently. In that case, these roots of higher plants, they make a pact with fungi. They make a pact with fungus and they let fungus live inside their body and in return ask them to absorb water and mineral for them. So fungi gets the shelter, prepared food, whatever the leaves will prepare food, fungi will enjoy that also. It has got a home sweet home. In return, plants are simply asking, perform the role of root because my root is not capable enough of absorbing water and mineral. So fungi is absorbing water and mineral for the plants, in return getting the shelter and prepared food. Thus, it is again a mutualistic association. And this mutualistic association is known as mycorrhiza. So, what is mycorrhiza? It is a mutualistic association between roots of higher plants and fungi. Why? Because benefits, या role. Just a second. So in case of fungi, so in case of mycorrhiza, what is the role of fungi? Simply, the role of fungi is to absorb water and mineral for the plant. Correct? And what is the role of plants? The role of plant is to provide shelter and food to fungi. And why is it important? Why plants need fungi? Why can't they themselves absorb water and mineral? So the simple reason is their roots are inefficient to do so. They are woolly, they do not spread deep, and they lack root cap. Am I clear? So these are two different organisms who also did not find place in Whittaker's classification system. So with this, I hope the entire chapter is clear to each and every student. What we have done in this chapter, we have discussed about first all the classification systems, 2 kingdom, 3, 4, 5, 6, and then we started each kingdom separately. You did kingdom Monera, including Archaebacteria, kingdom Protista, kingdom Fungi. Plantae will be taught in the next chapter. Animalia kingdom will be taught you separately. And then we also did all those organisms who did not find a place in Whittaker's classification system, like viruses, lichens, and mycorrhiza. Hope this entire chapter is absolutely clear. Focus on NCERT, read NCERT for solving all the questions. Now, some questions I have also brought for you all, which I write now. Okay, so yes, some questions we will also do, and then we are going to proceed further. Finally, I hope you all have understood the chapter, the entire chapter of biological classification, and now to ensure that I have 1 or 2 questions which I am going to discuss. These questions I have picked up from previous NEET exams. I hope they are going to help you in revising the concepts which you have studied. Let's see the first question which is on your screen. Who was the founder of 5 kingdom classification? Your options are Aristotle, Whittaker, Copeland, Haeckel. So I don't think you need to go for the options. You already know the answer, that 5 kingdom classification was proposed by R.H. Whittaker. Thus, the answer of this question becomes B. Now, if I talk about the remaining given options, then Aristotle, he has not given any classification system. He is the scientist who actually gave the first scientific approach for classification rather than giving a proper kingdom classification. Third, Copeland. Copeland founded the kingdom Monera. He actually gave the 4 kingdom classification. Haeckel coined the term Protista and proposed 3 kingdom classification. So according to the question, answer becomes 2, that is B, R.H. Whittaker. Hope you have understood this question. Moving ahead, next, heterocysts in some cyanobacteria are specialized for. So you should know that what are heterocysts. Heterocysts, my dear students, these are specialized compartment found in some bacteria like nostoc or anabina. These specialized compartments in the filamentous cyanobacteria helps in nitrogen fixation, as these heterocysts create an aerobic condition inside them. They lack photosystem too. They have thick wall around them. There is no exchange of oxygen inside. Heterocyst conditions are anaerobic, so that nitrogenase enzyme inside the heterocyst remains active for the process of nitrogen metabolism. Therefore, these heterocysts in cyanobacteria, in some cyanobacteria, are responsible for nitrogen fixation. Hence, the answer of this question, you all know, becomes A, that heterocysts in some cyanobacteria are specialized for nitrogen fixation. Hope you have understood this question, and with this, with my whole heart, I would like to end my session and this chapter, biological classification. My dear students, if you face any trouble in understanding any of the concept, you can mention in the comment section, and please do remain connected, as the next video soon will be uploaded for the chapter 3, plant kingdom. Again, very, very important topic for NEET point of view. Thank you all. Stay happy, stay focused, keep on working hard. Thank you so much.